I'm declaring that we do have a quorum present for our meeting today. The purpose of this meeting is to act on important business related to the practice of natural medicine in the state of Colorado. The board will make its decisions on agenda items today based upon written materials provided for its consideration in advance of this meeting. The public is welcome to listen. The public should not address the board unless requested by the board. As a friendly reminder to board members, please note that some of the material contained in the agenda packet may be deemed confidential. In addition, the board should not discuss legal advice previously received or seek legal guidance in an open session. Such matters will be moved into an executive session for discussion. Members of the public will be asked to leave the meeting during the executive session. After an executive session, the board will vote on agenda items discussed in the executive session. When we do a roll call for board members and non-voting participants, please indicate your pronouns, and if possible, please add your pronouns to your Zoom title. You can do that by clicking on the three dots above your image box and changing the panelist appearance. With that, I'll take roll. Dr. Brad, Connor, me, I'm present. Skippy? I didn't see Skippy. All right, we'll come back to that. Billy Wynn.
Here.
Dr. Sue Sisley.
Sorry.
Is that you there, Sue?
Yeah.
All right. Lundy? I didn't see Lundy in here either. So right now our quorum, Sam's here though. Great. Welcome, Sam. Our quorum is, it consists of me, Billy, and Sue. So we'll proceed. If others join, we'll have them introduce themselves when they get here. A few housekeeping items we get started. I'd like to ask everyone to turn off their cell phones or turn them to silent. If you have to leave the meeting at any point, please let us know so we can reflect that in the minutes. The same goes if you return. Also, please do not use the chat function if it's enabled for you on Zoom. And with that, I'll turn it over to the program director to introduce staff. So Lori, I'll turn it over to you now.
Thank you very much. So the staff that is here, well, first I'll start with myself, Lori Bratton. I'm the Natural Medicine Program Director. We have Sam Bairami, who is the Senior Advisor. We have Amanda Weirs. Amanda. Amanda's running the background. She's running all the Zoom meetings. Thank you. And she works in centralized services. And I do believe that I saw Monica Sutherland here, who will be taking notes for us. She's a policy analyst with Dora. And if the AG would like to introduce themselves.
Good afternoon. This is Brian Urankar with the Colorado Attorney General's Office.
Thank you.
And then I don't see anyone from DOR here. Is there a representative from DOR?
I believe Allison Robinette or Kyle is here. Davis.
No, they're the hands up. Thanks, guys. I apologize if I jumped out of the line on that. Lori, did you want to introduce DOR staff?
Well, I would love to introduce the DOR staff. Can you raise your hand again? Elizabeth, we have Kyle Lambert, correct? And Davis Grant, nice to meet you both.
And
thank you.
DOR is going through MED rulemaking, so most of the team that's working on natural medicine is going through that process. I believe, Kyle, you're on. We can't hear your audio, but
can you hear me now?
Yes.
Yes.
Okay. Hey, everybody. Kyle Lambert, Deputy Senior Director with Department of Revenue's Natural Medicine Division, joined by my colleague Davis Grant. And yeah, we're in a bit of a divide and conquer. So Allison Robinette, myself, Davis, Grant is also supporting the conversation from DOR today.
Great. Sam, just a quick point. You changed the name in B of 3, but you didn't change the name in 3. So if you also want to change to Lori's name on point 3 in that agenda for delegation of authority.
Yes, I think it should say that in the script now on. Oh, I see what you're saying.
Yeah.
That, you're right.
2 places. Yeah, just to make sure. Yeah, and I know that's not the same script that people are looking at. So now it's just me and Sam having a conversation while you listen to us, and that's fun too. But I just want to make sure that we're on the same page. All right, so next we'll move to the first piece of business, which is to a review of the meeting minutes from our last meeting, which was held on September 20th of 2023 at 1 p.m. We'll have Amanda scroll the minutes for us now and have you all review. After review of the minutes, I'll call for a motion to handle both pieces of business, which will be to approve the meeting minutes and to delegate authority of signature to the director, to the program director, Lori Bratton. So once you feel like you've reviewed the minutes, I'll wait to hear for that motion in a second.
Well, I move to approve and designate Sam.
We would be designating Lori, but thank you.
Oh, sorry, Lori, sorry.
Yeah. It's tough. We're all getting used to the new roles, but yeah. All right, so thanks, Dr. Sisley. Do you have a second? I think Billy is the only one who could second it, right?
Second.
Thanks, Billy. So all those in favor of approving the minutes and delegating authority to sign those to Lori, say aye. Aye.
Aye.
The motion carries. We'll move now to the first agenda item, which is to introduce the non-voting subcommittee participants. When I call your name, please just introduce yourself. Let us know where you're working and give us your pronouns. And I'll start with Scott Hansen.
Scott Hansen, Quality and Laboratory Ops for Ashta Biosciences out of Littleton, Colorado.
Pronoun, Scott?
He, him.
Thank you. Dr. Duncan McKee?
Director of Pharmacology at Bud Mary's MedPharm Research, he, him.
And did I say it right this time, Duncan?
Yep, Duncan Mackey. You're good.
Duncan Mackey. All right. I always want to say McKee, but Duncan Mackey. Nathan Wilkes.
Hi, Nathan Wilkes, health data and health data security expert. And for some reason, it's telling me I can't turn my video on thanks to the host, but oh, there we go.
And if you don't mind pronouns.
He, him, all the H's.
Thank you. Dr. Ken Kassenbrock.
Hi, I'm a professor emeritus at Colorado State University in Fort Collins. Pronouns are he and him.
Thank you. Hamilton Pevick.
My name's Hamilton. I'm the president of the Western Colorado Mycological Association and founder of Hamilton's Mushrooms. My pronouns are he, him.
Hamilton, can I ask a quick question for clarification? In my notes, it says Mycological Society. Is it Mycological Association?
It is.
Right.
WCMA, WICMA.
All right. If we can get that change reflected in the notes, that'd be helpful. Thanks. Dr. Peter Hendricks.
Peter Hendricks, professor, clinical psychologist at the University of Alabama at Birmingham, he, him.
Thank you. Daniel Story.
Daniel Story, I'm CEO of CMDC Labs in Longmont, Colorado. He, him.
Thank you. Skylar Sepik.
Skylar Sepik, he, him, co-founder of Microsoft.
And Skylar, I'm just noting that your name in Zoom is spelled different than mine. The name I have in the notes, it's SEFVIC, and then in the Zoom it says SEF. I just want to make sure you're spelling correct.
That is correct, without the V.
Without the V. All right. Again, so if we could get that reflected in the notes to get the V removed. Heather Krug.
Hi everyone, I'm Heather Krug. I am the regulatory program strategy for the State Public Health Laboratory at CDPHC, and my pronouns are she, her.
Thanks, Heather. I promise I'll start remembering that it's Krug. I keep saying Krug and I apologize. Dr. David Kaha.
David Saha, managing director. Oh, this is all good. Yeah, managing director at Science Lab. He, him.
Thanks. I apologize for mispronunciation. Dr. Andrew Novick. I'm having a rough day. I apologize, everybody. Is Andrew here?
Absent.
We'll go to the next one. And I apologize in advance if I get it wrong. Dr. Nigam Arora.
Oh, you got it perfect, actually. So, yeah, my name is Nigam Arora. I'm a PhD organic chemist. And if you could update on here, my affiliation is with Marku and Aurora, which is a boutique consulting firm that my partner Jehan and I have been running, specifically doing safety work and pharmacology work around psychedelics. So it'd be great if we could add that in there.
Can you say it one more time for the record?
Yeah, it's Marku and Aurora, MARCU & ARORA.
Thanks. We'll get that reflected in the notes.
Appreciate that.
Next is Dr. Jehan Marku.
Hello, I'm Jehan Marku, PhD with Marku and Aurora. Pronouns are he, him.
Oh, yeah, did we get your, Dr. Aurora, did we get your pronouns?
He, him. Oh, sorry.
Apologies. Yeah, he, him for me as well, and I'll add that next time I log into Zoom.
Thanks. Thanks, everybody. Colton Brook.
Yeah, hi. I'm Colton Brook. I'm the laboratory manager at Altitude Consulting. Pronouns are he, him.
Thanks. Ian C. I don't know if Ian's here.
He's at MED rulemaking.
Right. Same for Allison, right? So, and Brandon?
He's at MED rulemaking as well.
All right. And I think we got Kyle and Davis already, right?
Yep.
All right. So with the introductions, then we'll dive into the consolidation of recommendations that I prepared and see if there are any comments or adjustments to these recommendations. And just to remind everyone who isn't familiar with our task, all recommendations are draft. When they are fully consolidated into a report of recommendations, the Department of Revenue and the Department of Regulatory Agencies will file their rulemaking notices for all draft rules based off of the board's recommendations. Draft rules will be open for modifications, public comment, and feedback before they are finalized. The focus today is to go over what we currently have and to identify the gaps so we can work on addressing gaps. With that, let's get going to the first section, which are the draft recommendations for testing. And this is based off of the conversations that we had in our September meeting, where I thought we ended up. And so the draft recommendations for testing that I put forward for us to discuss here are: potency testing should be conducted for all batches of psilocybin using best practices for the testing of psilocybin and psilocin. Potency testing should include testing for psilocybin and psilocin. A batch should be considered any single flush, regardless of size and weight, and any new flushes would be considered a new batch. So I'll stop here with this draft recommendation number 1 and see if there are comments on this. Davis.
Yeah, hi. So I had an idea concerning basically the quantitation of total active psilocin as a potency measure. And I can share my screen and kind of present an outline of my idea if that's, that would be helpful. But basically, psilocybin is a prodrug that's converted to psilocin, which is the active form by dephosphorylation by alkaline phosphatase. And that mass conversion is from 284.25 grams per mole to 204.27 grams per mole. That's psilocybin to psilocin, which if you divide by those molecular weights, that conversion factor is 0.719 basically. And in marijuana,
Davis, can I put you on pause for Davis? Can I interrupt you for 1 second? Sure. Is it possible for Davis to share a screen so we can see those numbers? If not, it's okay. I just wonder if it is possible.
Davis, do you have that option right now?
I do, I believe. Yeah. Okay. So
thanks. That might be easier if we can see it. Yep.
All right. So basically, we're talking about dephosphorylation, so loss of this phosphate group due to the action of alkaline phosphatase, and basically what that means is that for every milligram of psilocybin, it will be converted to 0.7186 milligrams psilocin. So in marijuana, we have the same issue where HCA is the endogenous cannabinoid in the cannabis plant material, and in our sort of calculation of total active THC, we use the same conversion factor based on the decarboxylation of tetrahydrocannabinolic acid to 9 THC, which is the active form. So you can see that mass loss here. There's a carboxylic acid group right here. That's lost. And basically what you're talking about is a 0.877 conversion factor. So if we wanted to quantify total active psilocin, we could do that using this conversion factor, where we could say percent psilocybin or milligram psilocybin or milligrams per gram psilocybin, and use that 0.719 conversion factor due to the mass loss from the dephosphorylation here.
So, David, can I interrupt you with a quick question?
Sure.
So you're suggesting that for purposes of testing and or for purposes of labeling that we would label only the psilocin, not psilocybin and psilocin?
Not necessarily. So again, I'm kind of mirroring the same situation in marijuana. We label THCA, so percent THCA. We label percent THC, and we label total THC, which is an active form of what is potentially there once all the THCA is converted. So I don't know that this model would work or be applicable in the same manner in psilocybin mushrooms. I just wanted to propose it, and I would support that we label both psilocybin, psilocin, and perhaps total active psilocin, which would incorporate both.
All right, David, there's a number of hands up. I'm assuming they're either comment or questions on the things you're presenting now. I'll go to Nigam first.
Yeah, so quick one. I support what Davis is saying. I think that is highly reasonable, and I completely agree with it. The second comment I was just going to make is about the language around the batching. A single flush of any amount, any weight. I think there's some risk in that. If there is a humongous batch, you're going to have inconsistencies. So there may be merit to limit, like, I think just having any amount as a single flush is a little bit ambiguous, and people may seek to take advantage of that. So that's my other comment.
Yeah, and so, Nick, I know this is I think this is the first meeting you've attended as a non-voting participant. Generally, what we do is we start out. I like to start out with a set of rules so we can discuss them and figure out what's best in that practice. And so I started out with sort of the least conservative approach, and if we move towards more conservative, that would make sense. Do you have a quantification where you think you might put some limits on that?
I would have to get back to you on it. I also am curious I would have to get back to you on it, and I'm curious if there's like certain regs around the cultivation regs, which I haven't read in great detail recently. So, but I could get back to you on that and seek some other opinions.
Okay, so just as a way of orienting, we're writing the cultivation regs, so we're doing all that. We're making the recommendations at least, right? So the way that this proposal came about is from previous meetings with discussions with other experts around what, for instance, what the limitations are in Oregon, right? So in Oregon, there's some pretty tight limitations that actually dramatically increase the cost of testing and are likely not realistic. And so I wrote the rule to be on the opposite end of that. And so if you wanted to look into it and come back in our meeting in November with a proposal on limits for weight and size, then we could entertain that. But I appreciate your point of view on that. Thanks.
Sure. Yeah, and I appreciate you sharing the details on the prior things.
Sure. Ken?
Sure. So the comment about the equivalent in molecular weight in psilocin and psilocybin is useful. My thought, though, is that most people and all of the published studies people are used to thinking in milligrams of psilocybin rather than psilocin, even though psilocin is the active compound. So I'm a little bit wondering if it might be better to do the conversion the other way, to list both numbers, psilocybin content, psilocin content, and convert it backwards, potentially, say potential psilocybin equivalent rather than psilocin. I mean, it's the same information, but presented in a different way that people may be more familiar with or receptive to.
Sure. Davis, did you want to make a comment about Ken's suggestion?
Yeah, I mean, I totally support that, and I recognize that the sort of situation in cannabis and psychedelic mushrooms is the opposite in terms of the vernacular understanding of psilocybin is that it's the active ingredient. However, that's not the case, and the flip side is true in THC. We think of THC being delta-9 THC being what is endogenous in the cannabis plant, which is wrong. It's THCA. So I do think that proposal makes sense, and we could absolutely do it the other way around.
Is this an or question, or could this be an and question? Could we do both?
I suppose so. I don't know that it would add value.
And I'm just asking the sort of the devil's advocate question. This idea. The one thing we want to avoid and Davis, what comes to mind for me with looking at the conversion is that because the number for psilocin will be smaller, then people may be smaller than the number would be for psilocybin, then people may be taking more because they think they need more. Ken, is that the point you're trying to make?
Yes.
And so I think those are good things to consider. Thanks. I got some notes on those. Skylar?
So yeah, a couple of comments. I do agree with Ken there that, and especially from like a sales standpoint, selling directly to practitioners who may not be educated, about the conversion rate and all that, and then how to charge. We know in Oregon that they're charging per milligram of psilocybin, and I guess the market would have to figure that out on their own. But I think it's also important to note that in the psychedelic mushrooms, the conversion happens naturally through degradation. It's appearing to be around 5 to 6 weeks. So there's some things to consider, as far as like accuracy over time. Mm-hmm. The psilocybin will convert to psilocin, and then that psilocin will degrade after that. So those numbers change quite quickly in reality, depending on how they're stored and packaged. And I think that's something that should definitely be considered.
And so what we'd be looking at, Skylar, then, at the initial testing for potency, if we're going to label what that initial potency is, the maximum potency that the product could ever have, right? But it would degrade over time. And we don't have any, the degradation is not a constant, right? It depends on storage and moisture and oxygen exposure and things like that.
Yeah, absolutely. And I don't see any problem with having both numbers on the packaging. I would imagine that most practitioners would prefer to dose as accurately as possible, and so that potential psilocin number, I think, achieves that. I think that is the best number they can use to dose appropriately.
It brings to mind for me another question, and Skylar, you can answer this, or anyone else can answer this as we go. But it would make sense then, or should it make sense then, that we should put the date that the potency testing was done on the label as well, so that people know when that maximum dose was possible?
100%, absolutely. The date of testing is critical, and I'm sure as time goes on, we'll have some more standard models of how quickly they degrade. And we'll also, I know I'll personally be doing testing on packaging to see how much we can negate that. But that will have to happen later.
Sure. And going to just real quick to MED, back to you, Davis. I don't know off the top of my head, is the data potency testing for THC currently included in the label?
No, I don't believe so. I don't think the data is included on the label or required. There is an expiration date for or use by date, best if used by, to capture sort of potential degradation over time. This may be more sensitive in psilocybin mushrooms. I gathered that it is based on the literature that we've already gone over. So this would kind of capture the maximum available psilocin equivalent, at one point in time. Stability studies, I think, should be required and used over time to gather a shelf life and sort of, study the degradation over time of psilocybin to psilocin and psilocin to some other non-active metabolite. So, those are generally my...
But my understanding is that THC is a much more stable compound, right?
Yeah, I would say so.
All right. Dr. Sisley.
I was going to I think I may end up reiterating what Skylar, but I just want to emphasize that our work with psilocin in our lab under a DEA manufacturing license, it confirms exactly what you're describing. Psilocin is a super unstable molecule. It is very hard to maintain, even when you're preventing light heat, moisture exposure, it's still really easy for that to disappear. So I hope that what you're learning from these comments are that we have to make stability testing a priority in this program. I hope that you guys will figure out a way to empower these labs to work with, licensed manufacturers and labs early enough that they can start doing the stability testing as early as possible, because this is going to be very confusing later.
Yeah, Davis, can I get you to stop sharing? And Amanda, can we put the recommendations back? The draft recommendations back up?
Yeah, it looks like number 4 was the one about stability testing.
Yeah, so that came about based on a couple of things, Dr. Sicily, that we've been talking about at the various meetings that we've been having. And the reason I wanted to put it in as a recommendation is because my understanding is that Oregon does not allow for retesting, and so I wanted to make sure that we were recommending that we do allow for retesting, and we might need to put a statement in there that I think the one concern, I think my understanding was the reason that Oregon didn't allow for multiple potency tests is they didn't want people to be shopping for the best result to be able to label with the highest potency. And so I think the idea of, finding through the data process, finding how often degradation testing or stability testing should occur, I think is something that's still an open question. And I don't know, do you know of any data out there that suggests how often we should be testing potency?
We're attempting to create that data now. We're doing testing on tons of different strains of cubensis and hoping to publish it, make it all open source. But it's a long process, and that's why the sooner we can empower the cultivators and labs to start working together on this and legal. Great.
Thanks, Nathan.
Yeah, I think I would agree with all of the put all the numbers on that you can get in the testing. There were some comments whether you're doing, the potential of total psilocin versus total psilocybin is probably less of an issue, because in the current environment, users that are using the substances, not everybody's getting the substances tested. The amounts per strain for different batches, different grows, there's a lot that goes into it. And users are typically talking about dried weight of products in grams or whatever. But this is an educational thing. As all of this is getting tested, then we're going to be showing these component parts. And I believe there were comments in the last meeting that suggested even including the other tryptamines as well, baeocystin, et cetera. And that sounded like it was a normal piece of testing. So I'm all in favor of maximum education, maximum consumer awareness, putting them all on there, and whichever way you do it, you say, well, this is the effective amount of psilocin plus all the other things.
Nathan, is that a well-known and finite list? Pardon my ignorance of the tryptamines.
There was, I'm going to defer to the mycologists on here and those that do testing, but I think it is.
So, and any of the mycologists want to make a comment on known tryptamines and how many are we talking about? If we want to label all those,
it's just a handful, I think.
John, you have your hand up and you were just nodding. Do you have a comment on the number of?
Oh yeah, I mean, I think, are you referring to baeocystin and norbaeocystin?
Yeah,
norpsilocin. Yeah.
Yeah, I think those are good to include.
So I just trying to get a sense of how many things, and again, and I don't know if any of the labs are present to talk about. Is that standard part of testing?
No, it's not. I'll jump in. FDA doesn't require we test anything but psilocybin and psilocin right now. We don't know if any of these other alkaloids, though they're interesting, if they are exerting any clinical effects. So just to keep costs low for patients, I would say don't go beyond those two. But certainly for academic purpose, if labs want to test for those, go for it and publish all that so we can learn. But I wouldn't want the state to mandate that.
And here, what I'm really focusing on is what's going to go on the labels. So, we might still mandate that those things get tested for. We might still recommend those things get tested for, but maybe we're not putting them on the label. But it sounds like, so you're even saying we shouldn't mandate they get tested for to keep the cost of testing lower?
Yeah, I think it's going to be tough. Those reference, right, those samples are going to be super costly to get all the different reference material for those different alkaloids. Not all of them are even available yet. So I would just say focus on the two. FDA even admits that those two are the only two they care about. And then let labs that are ambitious enough to, work on doing all the chromatography for those different alkaloids. They can, document their findings and publish papers on it. But I think none of the public is really going to be able to probably know that. I mean, the lay public won't know what those alkaloids mean, and we don't know if they're exerting any clinical effects.
Yeah, I think it's the question of should versus may. So should would be psilocybin, psilocin, but if they do those others, they can certainly add those to the label, I think, is the appropriate.
And they're typically present at much lower concentrations. This system nor system, it's 10% or less of the psilocybin content. So
thanks, Ken. John.
Yes, so similar comments. Again, I am in favor of including more information about ingredients, but I also want to maybe just phrase this from a public health perspective. What's the most important value? Because if we're tracking, let's just say like adverse events into systems or ER visits, what do people put into the electronic system to look for? Is it psilocin? Is it psilocybin? So I think we need to have a value, whatever that represents, that is a single thing that can be pulled out and tracked for consumers, for public health, because, like the FDA spells it, with an E at the end. So, if you're searching the same term in different databases, you won't find it. So I think it's important to just think about, I guess that's my question. What would be the most important compound or description for this sort of data tracking?
Yeah, and so I think those are good points. And I think one of the things that came up as we were looking at how we were going to do this, even looking at the rules in Oregon, right now the rules in Oregon only require labeling of the amount of psilocybin. They don't even require the labeling for psilocin. And so, I think that's why we were leaning towards the labeling of both. My primary concern relates back to, I think, the point that Ken was making. I liked Davis's equations, but I don't want people to think that because the number is lower, they need to take more. And so I want to make sure that the public understands what the potency number is. And it sounds like the public may have a better understanding of what the psilocybin number is. So it may be that it's a better recommendation to reverse the equation rather than going with providing the psilocin, providing the total psilocybin, which also would include the psilocin that's present. Did I just make any sense at all? I got lost in my own comment.
I think that made sense, putting like a total psilocybin content on package.
And I would even suggest it be labeled as potential psilocybin equivalent, right? Because psilocin is so unstable. So this is it. If maximum, right. If everything is going to convert, then this is what you could end up with. But in fact, you'll probably lose some.
Right. That's why I was thinking about this idea of the maximum potency, and then the date that it was tested, so people would understand at least when it was that potent. Right. That's why I was thinking about the date stuff. Next thing I see is Colton's.
Yeah, I just wanted to jump in. I do testing on many of the compounds found in natural medicine, and so I can just tell you that right now our panel consists of 12 different alkaloids. We do find others than psilocybin and psilocin. Again, I think it was mentioned that we don't necessarily know what the effects are of some of these other compounds, but if there is data that does come out that shows that some of these other compounds, excluding psilocybin and psilocin, have effects, that may be something that you do want to consider requiring testing for.
Colin, do you know the cost difference if you're testing for those alkaloids versus not testing for those alkaloids?
Yeah, so the cost difference to run is not going to be very much. It's going to be an upfront cost, obviously, just to buy the reference materials. It also depends on what tier of reference material they are. So right now, I think there's 9 of the 12 or 10 of the 12 are CRMs or certified reference materials, which is one of the highest calibers of standards that you can get as an analytical laboratory. Vendors have continued to add more and more CRMs just over the last few months. And so I wouldn't be surprised if very soon all of the tryptamines and alkaloids that we can analyze for do have CRMs, but the upfront cost is essentially probably, you can look at roughly a couple $100 per standard.
Thanks. Daniel? You're muted, Daniel. I don't hear your audio. It doesn't show you as muted, but I don't hear your audio. Is anybody else hearing Daniel? I don't think it's just me. All right. But it's interesting because the mute sign is not on your name. There's just no audio. No, still no audio.
Output settings on the Zoom.
Still no audio. I don't know if you want to leave and rejoin. We'll come back to you. You'll have priority once I see your hand go back up, and we'll test your audio then. Sam?
Yeah, I actually was wondering kind of on the cost idea regarding like what is a recommendation for like a required thing to be tested for each batch. I'm interested to hear from DOR if for cannabis, if testing facilities are allowed to test for other things that the client might not have to pay for, but they just want to have data on when they're sending in product or anything like that.
Davis, did you put your hand up to answer Sam?
I did, yes.
Yeah.
So yes, the answer to that is yes. We require a panel of cannabinoids that is not inclusive of all of the cannabinoids that may be present. We also require some synthetic cannabinoids to sort of monitor the market for those. But there are laboratories that provide expanded potency panels that include, other cannabinoids that are not on the required list, and that's a service that they can provide that is not required, but apparently is desirable to some cultivations and manufacturers.
To answer your question, Sam?
Yeah, it did. I was just thinking on like a recommendation for like licensed testers to be able to offer that or service without it being like a hard requirement for each like batch.
Sure. Yeah, I think that makes sense. Danny, it looks like your audio is fixed, so I'll come to you next.
Is it fixed? Can you hear me?
Yes, we can hear you, yes.
Okay, great. So it's kind of jumping back in time and with the costs. Is it the plan to have a standardized protocol for testing for these things so that all the labs test them the same way?
That's my understanding. That our recommendation would be for standardized testing, yes.
Okay. And then, an additional part of the cost is proficiency testing, that every lab has, and how do we find that, being that psilocybin is, Schedule 1. So there, which I'm sure we could do, but, that kind of stuff does add cost to the lab. So, yeah, you can run all the stuff on the HPLC and it's not that expensive, but, from a lab standpoint, there are some other additional costs in there.
Yeah. Kyle, did you put your hand up to answer Daniel's question? No, okay. So yeah, we did talk a little bit at the last meeting around this idea about standardizing of testing. And my memory is that I'm questioning my own memory. That's why I stopped talking. My memory is that there are a set of standards that labs need to adhere to. Actually, I'll defer to the committee. Does anyone remember this discussion? If so, you could just start talking. Because I'm drawing a blank on what we decided. Duncan, did you put your hand up for this point or something else?
For this point, but I wasn't on the call last time, so no, I don't have.
Okay, no, you can add. Sorry, talk while I'm trying to remember.
Yeah. So for the standardization that occurs for cannabis testing is ISO, right? So all labs have to be ISO certified. You have to use all CRMs. So the cost for a test upfront. Yeah, sure, it's a couple $100 for CRM, but then there's also the research associate who has to set up all of the calibration curves whose time has to be calculated, the capital investment on the instrumentation, ISO's proficiency tests, which for cannabis, which is also Schedule 1, there's 2 of them a year. Based on how many samples you process, you either have to do 1 proficiency test or 2 every year. There's also any number of other added hidden costs that, all of your consumables and things like that. So pipette tips, the hours of documentation, because ISO requires, our we have two 5-inch binders for just our ISO SOPs and everything else that goes into being ISO certified as a lab. Talking about cost for one test, I would have to sit down and do quite a bit of math to figure out how much one test actually costs for one,
that's
one thing.
That's where I was going. And then, what is the ISO certification? 17025 or?
Yes, that's what we have.
That's what we have as well. But what is the state? I'm assuming the state's going to require that, but is 13485 okay? There's several different things, and then the protocol to be published, and then everybody has to abide by that, and then do the proficiency tests based on that. There is. It's like I said. I can run my HPLC for $100, but
yes. Well, and then there's mass recoveries. There's proving, like, for each analyte that you're looking for, you can accurately do it. So there's a bunch of extra work that goes into it behind the scenes.
Okay, if I can interrupt you, but most of all of those elements would be covered by having ISO certification?
They're all elements that would be part of your SOPs for ISO certification.
Yeah, but you would have to extend your certification. Yeah. So it's do you want to put this new protocol onto your certificate, right? Well, from a commercial standpoint, you want to, because that's advertising, right?
Heather, can I interrupt you for 1 second? Yes, okay. Heather, do you want to comment on this?
Sure, please. Well, I would say a couple things. One is, I think, yes, the state probably intends to require ISO 17025 accreditation because that is the ISO accreditation for testing and calibration labs. So that's the one that makes the most sense, but we certainly welcome any recommendations if there's a different standard or different requirement for laboratories that would be, in your opinion, appropriate for laboratories. I would just clarify and caution, though, some of the details of what was just being discussed in terms of what is required by ISO 17025. There are certain things that are specific, but in general, ISO 17025, it has broad requirements for what it is, quality management and technical requirements. The technical requirements are generally not detailed. The quality management, there are, again, generally broad requirements. So a lot of the specifics of how testing is done is up to the laboratory and or the method. So again, if you are doing pharmaceutical testing and we're following USP 467 for residual solvent testing, and that's your method, right? You should be following the details of that method, which have certain parameters of quality control requirements and things like that. But if you are a laboratory creating your own method, you could defend and justify to ISO why you are calibrating at a certain frequency or running controlled only every other day. That'd be kind of crazy, but there is flexibility in ISO. When you're talking about standardization and specification and what's required by ISO, again, there's a lot of room for the laboratories to, for lack of a better word, do what they want. So again, I just want to make sure everyone is clear that just saying ISO doesn't mean that we're going to tell labs to do things exactly the same way. And again, we don't always want labs to do things exactly the same way, depending upon technology that they have. So there's a lot of detail if we really want to talk about how labs do things. But that's not what ISO is. I just wanted to put that out there. It's broad requirements for quality management systems and demonstration of technical competence by a lab.
Right.
Not
but the protocol is going to determine how the accepted protocol is going to determine how we do stuff. So yes, I agree with that. So because you want all the labs doing it the same for the most part, so that you get the same answer, right, no matter what lab you go to.
Right, and so I think that, Daniel, your question is there going to, if I get this correctly, is there going to be a standard protocol that is part of the regulatory rule, the part of the rulemaking process that the testers are going to have to adhere to? Is that the question you're asking?
Yeah, so if the state decides to come in and audit my lab for testing, I'm assuming it's going to be similar to, and I don't know, this may not have been discussed, having auditors come in and audit the labs that are doing the testing. But, there's going to be, they're going to come in and do something similar to an ISO audit. How are you doing it? Is everything calibrated? Is everything, up to standard?
Yeah. Okay. So it sounds like we may make a general recommendation around ISO testing and then let the particulars sort themselves out. Heather, does that line up with what you were thinking?
I would say yes. And again, I think the concept of standardization of what should be standardized or what should be specified in terms of how labs do things, and that is very much in our mind and the process we will work through. Again, we want to see what recommendations this board makes, as well as we will be doing our own rulemaking process. So we will have that stakeholder opportunity as well to, iron out any other details. But, again, obviously, again, as I just said, we don't need to go into tons of details about this now. There are things I think absolutely need to be standardized and specified at a certain level, and then because this is a new area of science too, so it's not like we have a great reference method to just be like, this is obviously the gold standard, everybody should just follow this method. So we will work through that through our rulemaking process and over time, as well as we learn more about, again, what we're specifically testing for and the best ways to do that.
So Daniel, it sounds like the answer to the question. Duncan, do you want to jump in?
Yeah, I just wanted to echo what Heather was saying. The ISO is, it's a quality standard. So I come up with a method for mass recovery that shows that what I spike in recovers what I said I spiked in. Right. And Daniel does the same thing, but comes up with a completely different way to do it. Both can be defended as ISO.
Yeah.
And so they're both certified because you prove that the method actually works using the procedures that are set in place. It's not a standardizing one method for everybody.
But are we going to do that? Was kind of the question. Are we going to standardize it?
Right. So yes. So Duncan, even though you weren't at the last meeting, that statement just pinged my memory, which is to say, yes, that is what we're going to. The recommendation we're going to make is that individuals can develop their own methods. However, they have to show that what they're spiking is what they're showing with the method that they're using. That was
that would be ISO 17025 or any of these ISO standards, in fact.
So that's likely to be the recommendation that this committee makes to DOR, that's the procedure that be used. That was the conversation as we had it. And Sam, and so that was the recall of my memory as well. Thanks for the help, Duncan, even though you weren't there. Skylar.
I do want to kind of bring the conversation back to, what originally brought us down this testing road, and that's whether or not we test for all the alkaloids or the 6 major alkaloids or just psilocybin and psilocin. My concern is definitely more on the consumer side. Of course, I think we got to do everything we can to increase access to this and not, price anybody out from receiving this kind of treatment, and spending a couple $100 per flush is unsustainable. And I don't think that, the consumer would appreciate spending, $400 or what that may be for a single dose. I guess I would refer to Colton here. I know that them at Altitude, they're doing a really substantial sample analysis right now for an event next month in Denver. And I know they just raised their prices as well. But what do you think the cost to consumer would be or to manufacturer would be to test for more alkaloids versus just the main 2?
Well, do you want to go and answer that?
Yeah, sure. I can jump in. I just wanted to quickly go over the fact that proficiency testing for these alkaloids in mushrooms does exist. Accredited proficiency testing does exist. We just completed that a few months ago. Along with the ISO 17025, if there was any concern about whether or not these accreditation bodies would go ahead and go through audits for this sort of scope expansion, they do. We actually just completed the first ISO 17025 accreditation for these tryptamines. But yeah, going back to the cost basis, again, depending on whether or not your reference material is a CRM, you could probably bet on each standard being $200 to $300 for about 1 milligram of solution. So, if you have a 10 analyte panel, that's obviously an upfront, probably $3,000 just to get your reference materials. I guess the concern being if you're only testing the potency of every flush, I know it was brought up earlier that there's not an amount necessarily per flush, but for a lab to be sustainable, I'm not totally sure that one potency test per flush would really cover all of the costs that are associated with running a laboratory.
And Colin, when you say that, you're saying that in reference to even if we're just testing for psilocybin and psilocin, or if we're testing for all?
correct, even if it's just psilocybin, psilocin.
All right. And Skylar, did that answer your question?
It did. It was really not the answer I was hoping for.
I can see that on your face.
There's got to be a way that we can make this accessible and sustainable. Those numbers are just enormous, and I can't imagine passing that on to a consumer. There's no reason for them to go to the lit market. They would immediately go to the gray market. I just don't see the value.
No, yeah, I think that I'll tell you that the direction I'm leaning in terms of if I were to write a recommendation, it would be to require psilocybin and psilocin and say that maybe best practice is to test for the others, but make it an option, just to keep the costs where they hopefully within reason. Colton, how often do you think you'd need to do potency testing in order for it to be viable?
So from the shelf life studies that I've seen, it looks like about 3 months seems reasonable on the grower or consumer side. As far as how often I think that it probably depends on how large the flush is. This is something that we've seen that it really depends on size, how long they've been growing as far as potency. I mean, these things are factors that can vary greatly in a single flush. And so I think it's kind of hard to say at this point.
I'm going to see if I can ask the question a little more specifically. You were saying that if we only had potency testing once per flush, then that wouldn't be a viable model. Is there a weight or some other way you can tell me what point would it be a viable model? Does that make sense?
Yeah, sorry. Possibly you could possibly subsidize some of the cost with other analyses. I noticed that on the draft recommendations, it looks like testing for many contaminants occurs or recommended to occur randomly once per year. That is possibly another way to make it more sustainable for the lab. As far as just the potency, if you are just going to look at psilocybin and psilocin, the upfront cost will not be as much. But again, if you're running an ISO accredited lab, as was mentioned previously, there's many more costs than just reference materials.
Thanks for that. Nagum?
Yeah, thanks. Just as we're kind of chatting through this, I can't help but think about the unit economics of it, because when we talk about the cost of a standard, well, first I want to say I'm in full favor of keeping things affordable for the patient and the end user. I think that's super important. So as we think about that in the chain of events and the chain of costs that will occur, the unit economics really matter, because yes, these standards are expensive, but those numbers are big, but you're going to run a lot of tests with those standards. So I think that goes back to my initial comment about, okay, well, we're saying one flush is a batch. Well, that makes it hard to understand unit economics and understand what burden an individual grower would face to pay for testing, an individual lab would face, how it would trickle down to a consumer consuming a gram of dried mushrooms or 3 grams of dried mushrooms in a therapy session. So I think that even if it's just for the sake of example, doing some examples of, okay, a batch is 100 grams of dried mushroom, or a batch is 1,000 grams of dried mushroom, to help understand what are those unit economics and how does it really trickle down to the consumer for the cost of testing in general. I think that's important. And then someone else had made a comment about it's not just the standards, there's a lot of other costs in testing as well. But to really understand it on a quantitative level, we kind of need to get some hard numbers on it.
Thanks for that. Skylar?
I guess I just had another question for Altitude. And maybe helping me understand the difference between, this ISO model and what you're doing now currently for the event in November, with the hundreds of samples that you're processing there, what you're looking for and how the methodology is different, and where that extra cost is incurred.
Sure.
If he's still here.
Yep, I'm still here.
Okay, good.
I'm happy to discuss what we have just between you and me, Skylar. But just for the sake of this meeting,
I was like, you can all have that conversation offline. That'd be better for I think everybody involved.
Yeah.
Okay, I apologize. Sorry.
No worries. No worries. Davis?
Yeah, I'd just like to provide some context on the cost of adding additional analytes to a test. For example, in marijuana, we require testing for a certain panel of pesticides, and we required testing up until July 2023 for only 13 analytes. We've now expanded that to just under 40. So you're looking at a 3 times increase in the analytes that we're actually requiring for that test, for compliance testing. And we have not seen a drastic increase or are not aware of much of an increase in the testing cost for pesticides and marijuana, even after tripling the number of analytes that we're looking at. And just to provide some additional information, we're seeking to expand that panel from right around 40 at this point to 99 in July of 2024. So the testing for 5 analytes versus 10 analytes does not cost twice as much, and I think that needs to be the point here. And as Nigam mentioned, that cost will be spread out via the number of tests that are performed and the size of the batches that those tests reflect the results for.
Right. So, if possible, what I'd like to do is have a more of a specific discussion around point B of subpoint B of point 1, which is right now, as written, the recommendation that is a batch should be considered any single flush regardless of size or weight. It seems like we might want to put some limitations on that, or at least it seems like some of you may want to put some limitations on that. And that might help us move a little bit forward in terms of determining what the cost might be passed on to the consumer if we had some idea of that. Our previous discussions, the reason I wrote it this way is our previous discussions were because the Oregon regulations were quite strict that drove the cost of testing up. And so I just wonder if people have recommendations around what a batch should be considered. Nico?
Oh, thanks. I still would need to do a little back-end work to make a solid recommendation. But I was just going to comment that there may be a mechanism where there could be a batch size defined for testing, but then separately there could be let me just say like a concrete example, right? Let's say that you say 100 grams of dried mushroom is a batch for testing, just so that we're not getting humongous variability when you're testing 1,000 or 5,000 grams. There's going to be a lot of variability. So just for the sake of testing, you could define a batch as one thing, but then you wouldn't necessarily require a grower or a purchaser to test every 100 grams. I don't know if that makes sense. I guess I'm just saying you could get a better level of specificity in your testing without necessarily requiring that amount of cost is passed on to the consumer. I don't know if that was confusing or if that makes sense.
I'm somewhere in the middle between it's confusing and it makes sense. I'll come back to you in a second. Davis?
Sure.
I think I can shed some light on that. What we do in marijuana enforcement is basically we have required test batch increments that correspond to the total harvest batch or production batch size. So if your harvest batch is 1,000 grams, then you're only required to select so many test batch increments. These would be random samples pulled from throughout the batch and would be put together to make a composite representative sample of the total harvest batch being tested. So we could have and we do in the regulated marijuana market have rules that dictate how many test batch increments are required and what those test batch increments, the mass of those, which is, I think, half a gram or a gram. But it could be applied to different scales of flushes, is what I'm trying to say.
I like that language. Scott, were you going to comment on that specifically, or on this idea of weight?
Yeah, I was. I think it's important to remember that, like, you know, the variability between potency is going to be probably the closest in a per flush model. Now, it's really difficult to say how much a flush is going to produce as far as weight wise. Like, you could get a single mushroom that grows more than 100 grams in its wet weight. So I think it's really important to just consider the product here. Like, you know, I think the per flush model is the best we can do as far as, like, to be accurate. There will be variation from mushroom to mushroom, but it's going to be really hard to quantify that, I think, in a meaningful way.
So you're not suggesting any weight limits then, if I'm hearing you right, Skylar?
Yeah, I mean, I've seen 128 gram wet mushrooms, like, and you know, it wasn't the only one in the flush. So and there's, you know, it's just so much variability that it's going to be really difficult to set a limit. And the per flush model is the best I think we could do.
Appreciate that information, Duncan.
I was thinking, you know, within a flush, I mean, you can set tier limits, right? So in cannabis, you know, we don't have an idea of what every cultivar is going to produce weight-wise once it's dried and cured as a flower. You know, each flower has a different amount of moisture, the cure process, there's so many variabilities, but we do have sampling subsamples within each batch, right? Based on how much we get at the final product, right? So 0 grams to a kilogram, we have to send this many samples for testing. And then that's just on the biomass, right? So then once you talk about formulating a candy bar, you know, stabilizing psilocybin and psilocin in some kind of edible form, depending on the batch size, we have to send finished units for testing. Mm-hmm. And so thinking in that model, you can subset within each batch. So I don't mind the, like, per flush, but if you have a really, really large flush, then maybe you do have increments or tiers, so that way you can get more of an sampling idea of what the true potency of that flush was. Just as a suggestion based on what we do in cannabis. Yeah. And then also dividing, you know, manufactured products versus biomass, you know, a dried mushroom equivalent to a dried flower versus a candy bar, right? Or vape carts or something along those lines, where we make a big batch, we have to send more if the batch is larger.
Yeah, well, we also heard in our last meeting, you know, this idea that you could be testing the you know, those were not words, raw dried mushroom products, or, you know, if someone decides they want to blend the mushrooms together, if they want to grind them and put them into a powder, then you need to do potency testing on the powder as well. And so I think right now what we're discussing is, if from, you know, at cultivation to harvest, you know, what's the potency testing there. But I think we're also going to have some discussion about, once you move into production manufacture, you're going to have potency testing on the final product there.
Yeah, finished product testing. Yep.
Finished product testing, yeah. Nigam?
Yeah, I just following the protocol that was mentioned in the beginning, I have to hop off, but I'm going to do my best to hop back on shortly from my phone and keep at least listening to the combo.
Thanks for the information. Appreciate it.
Yeah, appreciate everyone.
Thank you. So let me just summarize what I've heard so far, where I think we're going, which is to say that if we're talking about potency testing for raw dried mushrooms after harvest, then the flush model looks the best, but we may want to put some upper limit on how much is considered for a total flush. That's where I sort of ended up. And again, I'm not saying this is what we're gonna eventually propose, but what I'm pulling from all the conversation we've had is sticking with the flush model, but maybe putting a cap on the upper limit. That not to say that you're gonna say it's a new flush or it's a new test, but if you have a flush that's over a kilogram, you need to have 2 potency tests done, is what what I think, what I heard, and again, I'm putting that out there so that people will tell me I'm wrong. Does that sound like a viable model to people? And I don't even know, I just picked a kilogram because that sounded reasonable to me. So again, I'm not a chemist nor am I a cultivator. If someone wants to tell me I'm wrong or stupid, please do so now. Skylar.
I'm not going to say you're stupid. No, I think what I understood was if it's a particularly large flush, that we provide a larger sample size, and that sounds fair to me. Yeah, if it's just a massive flush and we need a better idea of what the average is, we need a larger sample size, and I could understand that. Yeah, that's no qualms there.
Okay, Davis.
Yeah, so same as Skylar, I'm not going to tell you that you're wrong or stupid. That's certainly not my intent. But what we're talking about is basically statistical sampling methods that have been shown and developed to give a representative sample that's according to the batch size or the flush size. So for a 1,000 gram flush, you may have to provide a 10 gram test batch sample for potency. Now, if your flush was 2,000 grams, it may need to be, I don't know what the model says. We have a table in our rule book, but it may be double that, or it may be 1.5 times that, but it will be larger. And the purpose of that is to, like I said, create a statistically robust representative sample that reflects the potency of the entire flush.
Do those models exist? And this is not just to Davis, but to anybody. Do those models exist for mushrooms? Obviously, they exist for cannabis, but do they exist for mushrooms?
I don't think they're unique to any single commodity. I think they're broadly applicable to testing.
And then for the cultivators in the room, what is I mean, is there an average flush size? Like, can anyone give me numbers? Like, what do you usually see, or what do you usually plan for, or what do you usually expect?
I could say that you could expect anywhere from 20 to 32 grams per flush, maybe more after it's dried. I think that's the hardest part. That's something we'll have to consider too. Like, when is this being verified, this weight? If we're going to regulate, an upper limit before, a larger sample size is needed to be provided, when is it going to be weighed? Obviously, I think we'll probably go with dry weight, but
yes. How
are we going to verify that is another thing. Before it gets sent out to testing, those are precious days, as we know. They degrade quickly, so we want to test as soon as possible. So eliminating any lag time would be key. I could say, upwards of 2 to 3 ounces in a flush. I could imagine a 3 ounce dried weight flush.
Would there,
on the higher end.
Would there be a disadvantage to cultivating, trying to get a larger than 3 ounce flush? Like, is there danger in the cultivation process or to the quality of the grow, or like, I'm trying to balance the idea, because if we put this batch limit to the flush size, as I think Megan was pointing out earlier, that people might just be trying to get larger and larger flushes to reduce the cost of testing, right? But is that counterproductive to the production? Well, that's a lot of productions. Is that counterindicated for if you're trying to produce a quality product?
I think it gets more difficult the larger surface area you make your substrate. So I think there's a lot of different methods of how to go to fruitation. Right. But it seems kind of standard. The largest size you'll see is done in bins. The same bins you can buy from a Target. Mm-hmm. And that's generally the largest surface. Now I can imagine a larger flush. If someone were to make some custom made container for a larger substrate, surface area, but it does get risky. Your risk of contamination goes way up, the larger it is. So, I think it's possible, but probably not sustainable.
So in my imagination here is that most people are going to be cultivating in bags or in bins, right? You're not thinking people are going to be going larger than bins at this point?
No, and in fact, I look at some of the, commercial mushroom operations across the world, and a lot of them use bags. Believe it or not, I think they find it very, very easy to just mass produce through bags. But I mean, I highly doubt it. I really do highly doubt anybody's going to try to innovate some way to produce a larger surface area to get around a per flush regulation of some sort.
Right. And if we're talking about bags and flushes, are we talking about how do we our bins and flushes? How many bins are in a flush? How many bags are in a flush? Are we thinking it's one bag to one flush, or it's multiple bags to be a flush? That's another thing I was trying to consider.
Yeah, so I would, I'd say you'd have to consider each bag a flush. And now that bag will produce mushrooms 2 to 3 times, so that would be 2 or 3 separate flushes. I may have a few qualms with considering each flush a separate batch, especially for record keeping and numbers. But I think the bin model is definitely the best way to think about it. And yeah, each bin would be per flush. They had mentioned in Oregon that once the mycelium goes to grain spawn, and whichever method they're using, whether it be agar or spores or liquid culture, that's when it becomes a batch. And so I think that's important to consider because in most cases that will also be one bag. So one batch to one bag. In the bins, you might find that you might use 3 or more inoculants to colonize a bin of substrate. So,
the reason
I think
the reason we went the flush model, sorry to interrupt you, but the reason we went the flush model is this idea that there's not great literature that indicates whether the potency changes from one flush to another. And I think that downstream, once we get some of that data, if we're not seeing increases or decreases, significant increases or decreases in potency, we might remove that flush limiter. But the reason the flush limiter is put in there now is that we don't know how potency might change from one flush to the next. And I've heard competing evidence. Some people say it increases, some people say it stays the same. And so I think we need some data, but that's why we started with that, at least that initial per flush model. Does that make sense?
Yeah, it does. And I like that, especially a lot more than the 24-hour harvest period that they have in Oregon. But to consider it, and it's maybe just a struggle with the verbiage, but to consider it a new batch every flush, I'm wondering, do we have new batch numbers for that?
Right.
Because in every way, genetically, it's the same as the flush before, for the most part. So it just gets a little confusing after that for me.
Okay, thanks. Ken?
Yeah, I think we're kind of getting lost in the weeds and the details of cultivation, and I would like to just clarify a few things. Growers can do lots of different things. You can grow mushrooms like button mushrooms. I mean, you can grow psilocybin mushrooms the same way you do button mushrooms on a giant scale if you set that up. It's not difficult. Those are not difficult mushrooms to grow. It is true that people typically currently are growing either in special mushroom spawn bags or in these tubs that are talked about, but a batch may be setting up 10 of those at a time. A flush then is when you get a fruiting of mushrooms. Then in that same bag, you may get another fruiting 2 or 3 weeks later. That's a second flush. So flushes and batches were kind of confusing a little bit, I think. And I don't know that we need to be defining all of this at this moment. Yeah.
Ken, the lawyer's hand just went up, and I always go right to the lawyer when his hand goes up. Brian?
Well, it dovetails off that pretty nicely, and I think actually I'm getting a little lost in the weeds as well, but I think defining them is probably something you're going to want to do in a recommendation. Specifically, what's a batch, what's a flush, and I think just from a regulatory standpoint, the regulations become more flexible if you can scope those definitions in a way where you're not like you capture both bins and bags, but you don't say either specifically. So I would just encourage the panel to think of a way to do that, to kind of zoom out a little bit and use your defined terms in a way that covers, bins, bags, and whatever the next technology might be for cultivation in a couple of years or something.
So because the comparisons have been drawn so much so far, how is that determined either for Davis or for Duncan or anyone else who cultivates THC? How do you consider what's a grow in THC? Is it a plant? Is it a number of plants? What is it? Again, Davis or Duncan or anyone else who has that knowledge. Duncan, go ahead.
Yeah, so for us, a harvest batch is pretty much any plant that's harvested on that day. Gets lumped in. So it could be 3 or 4 cultivars. We might bring down Blue Dream, GSC, and some other cultivar.
Right,
but that harvest date.
So that explains the 24-hour model in Oregon, right? Yeah, that explains why Oregon uses that 24-hour model.
Yeah, because they do it by date, window, right? In Oregon, they do their harvest batches are the date that the fruiting bodies are collected. And so I think that's models what we do in cannabis, but I don't know if it's super accurate or helpful for mushrooms if we get to set it up and do it right.
I don't want to use a 24-hour model for mushrooms. We heard enough and saw enough in the Oregon regulations that it increased the cost of testing and it made a lot of things much more difficult. But I do think that having a definition is going to be important, and we're going to need to come up with that definition, whether that's per bag or per bin or a certain number of bins or a certain, and I know you can't put it on weight because you don't know what the weight's going to be when you're setting up a batch, right? But
and you can't do it as a bag being a batch because that's cost prohibitive.
Yeah, if you set up 10 bags with the same inoculant, technically those 10 bags should all be the same batch, right?
Right.
When it's a flush, right? That flush should be a batch.
So David, I'll come to you in 1 second. Duncan, but in your mind, what you're saying then is if it's at the time when you use that inoculant?
Yeah, that would make the most sense in my mind. I don't know other people's opinion, but if I have the inoculants, I inoculate 5 bins or 10 bags or whatever it is, I carry it out till fruiting bodies and I collect those, then flush, the flush. I think that would technically be, in my mind, that would be a batch or what you would test from.
Are there any upper limits? Are there any upper limits on inoculant?
I wouldn't have any opposing one way or the other on that, actually. I mean, I'm kind of open to it. I think you're going to get varying size grows, so I think you'll have a gamut of people doing different sizes of inoculants. So I can't speak one way or the other on that one.
All right. Dave.
Yeah, kind of going off what Duncan was saying, and some other folks, I also got a little bit lost in the conversation. But one thing I would encourage us to think about is, how facilitators or guys or whatever the terminology will be using this. So if we're doing testing per batch or per flush, will we be delivering, 1 unit, 1 flush for a facilitator? And then down the line, when we're assessing outcomes, are we going to have to be measuring each flush, kind of indicating which flush in the kind of outcomes report? So I was thinking if we could kind of, batch things in a bigger way, that could be more just easier to track down the line, like not just what the growers are doing, but also kind of what's happening at the outcomes level.
Thanks, Davis.
Yeah, so I don't have strong feelings about necessarily limiting the size of a flush or a batch. In the regulated marijuana market, cultivations vary in scale by orders of magnitude, and the way that we guarantee or do our best to require representative sampling and testing for all these different sizes and scale of harvest batches that may be produced is by that sort of rolling scale of between this and this amount requires this as a minimum test batch.
But there's also a date parameter. Sorry to interrupt you, David. There's also a date parameter that I heard from Duncan, right? Which is to say that the amount that's harvested in that day is also relevant here.
Right, exactly. And so our definition of a harvest batch is basically that which is harvested in a similar or limited amount of time, maybe being that 24-hour period.
Well, let me ask this based on the point that Ken made earlier. What if we flip the equation though? So how long is an inoculation window? Like rather than having a 24-hour window at the harvest, what if you had a 24-hour window at inoculation to establish your batch? Is that stupid? And I don't mind being told I'm stupid, by the way. No one's going to offend me if they tell me I'm wrong or I'm stupid. It doesn't bother me. But like, I'm just trying to think about how we avoid the cost crunch that we see in Oregon by having that 24-hour harvest window. If we move that 24-hour window or that time frame window to establish a batch to the inoculation stage.
That's not a terrible idea, to be honest. You could make an incredibly large amount of inoculant through liquid culture, and so you could. If that one liquid culture was to be considered a batch, you could produce many mushrooms off of one liquid culture. So I think putting that to a 24-hour period, like everything you inoculate in a 24-hour period, that's not a bad idea, but it may have to vary. Maybe you inoculate 3 different strains that day, and so you may want a different batch number for each genetic, profile. But yeah, that would seem reasonable and not difficult to implement.
Thanks, Skylar. Duncan?
Yeah, I think that's actually a pretty good suggestion. Outside the box thinking, by the way. That would be my only suggestion.
All right. Ken?
I wouldn't do it as a 24-hour thing. It's less. It's less than that. What people typically do is they make up a batch of inoculum. And it's called spawn, and spawn can be made in a bag or a jar. And then when it comes time to inoculate your bulk substrate, you use the entirety of that jar or that bag of spawn to inoculate however many things you inoculate, maybe 10 or 20 or 100, and that may take you an hour or two. That's a batch.
Is it reasonable to say then a batch is defined by the inoculation, not by the harvest?
Yes. Well, yes, that's the batch. The harvest is the flush. The batch is the inoculation.
Right. We would still have until we see enough data that suggests that potency is stable across flushes, I'd still want the flush limiter on the other side of that. Right. So again, I'm just trying to get my head around this. So if we say a batch is established at inoculation to first flush, and then second flush is second batch, third flush is third batch, but it's still based off that initial inoculation. Does that make sense to everybody?
No, it's confusing batches and flushes. A batch is when you prepare and inoculate. Flushes are, so a second flush off the same initial batch would be tested separately. But
so let me try it again. So let me try it again. So we would say a batch is the inoculation, and then potency testing would be required for every flush within that batch.
Yes.
All right, that's okay. So we've spent about an hour on this first point, but I'll rewrite the recommendation to look like that, and we'll review it again at the November meeting. And if we can, I'm going to push us through a couple more points, and then we'll take a break. Any objections to species testing once random per year, or anyone comment on this idea? That's where we landed at the September meeting, was that should be sufficient. A random test once per year for species. Does Jehan?
Yeah, I would say that I agree with that, but maybe adding language that or as appropriate, or if there was a big mix-up of species that could trigger an investigation. So just wanted to
there would be if a person fails the species test, then they would be required to have more repeated tests until they show that they're only growing cubensis.
And just to clarify, there's nothing else that would trigger an additional round of testing in this instance.
Not at this point, no. Okay. Brian?
Great. So,
to the point you just made, Dr. Connor, I don't know if you necessarily want to recommend a rule that's so specific that says, if you fail this test once, then you must do X number of tests. I think maybe a better way to approach it is make sure that the rule's crafted in a way that the regulator, DOR, has as an enforcement mechanism, or like a remediation tool, a way to impose X number of tests or whatever DOR thinks is appropriate, rather than saying as soon as you fail it, you're getting more tests. Because who knows? There could be very good reasons why they failed the test, or there could be malicious reasons why they failed. And I think you need the regulator to have the discretion to have extra tests if they need it.
Yeah, so I would update point 2 to say if a licensed cultivator fails a species test, then DOR would determine remediation. Does that make sense then?
That's definitely closer. Let me think about it, and I can maybe give you some draft language you could throw in there.
Sounds perfect. I'll wait for you to do that for me. Any other comments about point 2? If not, we'll move to point 3, which is a very similar point, but it just is this idea of testing for anything else randomly once per year. And again, I would add language, waiting for Brian to give me some suggested language around what happens if they fail. We'd have to put that as part of there. But as a starting point, comments on the idea of once per year random testing for other elements that might be present in the mushrooms. And here I'm thinking about heavy metals, pesticides, and mycotoxins. And if we want to add things to the list or take things away, again, I'm not a mycologist, so I just went with what was being discussed.
Microtoxins probably should read mycotoxins, which are toxins that molds make.
Not myco. Gotcha. Thanks, Ken. Others? Davis.
I just have a question about how the random testing would occur, and if the cultivation or whatever term they're going by is notified that, hey, you are required to submit a random test for these possible contaminants. Is that really random because they self-select their test batches? Just a thought, but just questions about how this would actually be carried out.
Sam, did you want to comment on that point?
Yeah, I would like to know how it's done in cannabis.
That's what I was going to ask too. Yeah.
Yeah, we don't require random testing unless there's an active investigation, and we can identify material, harvest batches, production batches that are present at the cultivation or at the production facility, and basically require additional testing for those. But that's during an active sort of investigation rather than just a rule.
But we don't have the analog or the same process whereby there's no species of cannabis that aren't allowed to be cultivated. Is that accurate?
Yes, that's correct.
Right. So that's why you wouldn't necessarily need random species testing, because anybody can grow any species they want, essentially, right?
I suppose, but I'm speaking more towards the heavy metals, pesticides, and mycotoxins.
Oh, okay. Bolton.
Yeah, I just wanted to jump in on the mycotoxins. I'm not sure if there's recommended storage or harvesting techniques, but you may consider just not performing specifically the mycotoxins randomly once per year, because based on how they're actually harvested, dried, and stored, I would think that may be a concern of basically mold or things like that growing.
I do think that there's a likelihood that we would make recommendations around storage, but we haven't gotten to that yet.
Okay, thank you.
Yeah, Duncan?
Yeah, and it's funny you guys are talking about these testings because we're doing a project looking at them in the cannabis market. Flower requires heavy metals, pesticides, water activity, and mycotoxins. Extracts, so distillates and dabbables, require residual solvent testing, heavy metals, and pesticides every batch. Infused products require homogeneity, mycotoxins, and heavy metals every production batch. So literally, we are continually testing all of our stuff all the time for heavy metals and pesticides in cannabis and mycotoxins.
Anyone want to comment on whether that level of testing would be necessary for mushrooms?
It's a huge overkill.
Yeah, I think it is in the cannabis industry, but that's another argument for another day.
You can argue that with Davis offline, I guess. But yeah, Yeah, Sue, I was wondering if you had a comment around this, because I know that you were definitely concerned. I remember you talking about being concerned about mycotoxins and heavy metals. I just wonder what your thoughts here are.
No, I feel good about the way it's written. I was just going to ask the group, the experts, if they thought anything, because I know we have to test for like micro, so rule out E. coli, salmonella. Do we need any of that, or can we leave that out?
Anyone want to comment?
I think it should be in there. Actually, I think specific testing for E. coli or microbial contamination should occur, because it's possible when you're growing wet mushrooms to have bacterial and or mold contamination, and you want to test for that.
Does that need to occur more frequently than what the schedule we currently set out, just randomly once per year, assuming that the tests are passed?
I think a good grower will make sure their product is fine. This happens with every edible mushroom that's grown. I mean, it's a potential concern.
Right.
You don't need to test every single day at mushroom factories, mushroom farms.
No.
You just need to make sure they know what they're doing every once in a while and that they're following good practices.
Yeah, I don't know if other people watch the well, it doesn't matter. I won't go to a TV reference, but I know FDA is overburdened with testing leafy greens and things like that for E. coli, right? We randomly we'll see that stuff. Davis?
Yeah, so this may not be all that palatable, but in terms of sort of random testing, something that could be considered would be a state basically conducted the equivalent of an off-the-shelf testing program, which would go randomly to licensees, collect samples, at whatever interval is determined, whether it's yearly or otherwise, show up, actually observe the collection of the samples, and those would be tested at our reference laboratory at the Department of Health and Public Health and Environment. And then besides that, in terms of microbial contamination, we require testing for STEC, which is shigatoxin producing E. coli, salmonella. Our mycotoxins are aflatoxin A, ochratoxin A, and B maybe. So there's a limited subset of microbials that can be present in marijuana, and I'm sure that there's, through further study, we can determine ones that may be likely to occur in mushrooms and produce a test panel that would reflect what needed to be tested for.
Thanks, Davis. I'm going to ask a question here specifically. So you had mentioned a process whereby you're adding things that are going to be tested for in cannabis. Does that have to go through a rulemaking process because of the way it's written? Is that more specifically, is that a legislative procedure to add more things that must be tested for?
I don't believe it's a legislative procedure. DOR is given the authority to require testing for certain, just at large.
Yeah, that answers my question. So basically, I don't want to specify exactly what's needed to be tested, because I want DOR to have the ability to change that list without having to go through a legislative process, right? But we can make the recommendation that both mycotoxins and microbiomes are tested for, and then DOR could define the list of what's tested underneath that, right? All right, that's what I was thinking. Okay. And then what the process that you described, Davis, was what I had in my head, which would be, but I would say the same thing. I don't know that, and I might look to staff here or even for DOR to comment. Does DOR want us to define what the process for random testing looks like, or can we rely on DOR to develop the random testing procedure?
I think that DOR can be given the authority through rulemaking to determine the test panels, the required testing, the frequency.
But even the test process, like the one you described, that off-the-shelf observation of sample collection testing?
Yes.
Okay, good. I was hopeful that you would say that. Thank you. I'm going to get us through point 4, and then we'll take a break if that's all right with everybody. So the one point I heard was potentially a 3-month window, at least what we're thinking right now. I'd like to put a time frame on the how often we should retest for potency based on best available knowledge that can then be changed as we gather more data. Ken, I saw you unmute, so I'm going to call on you.
Right. I think every 3 months is too often, and it will drive the price up quite a bit. You can store things in a freezer. I mean, everything has to be dry first, dry and then and protected from oxygen and moisture. But barring that, if you kept things in a freezer, for example, or even lower temperatures, it would probably be stable for a year or more. So I wouldn't go with a 3 month.
That brings up a wrinkle to all this, which is to say either we're going to recommend rulemaking on how mushrooms should be stored, or we might recommend different retest intervals based on how the cultivator is choosing to store the mushrooms. Did my point make sense?
Yeah.
And so if I'm going to store it in a freezer with desiccants, or if in a as dry as possible environment as possible, then maybe I can get to a 12-month retest window. But if I'm going to leave it in a jar on a shelf, then maybe I need to be at a 3-month retest window. Does that make sense?
Yeah, I'd hate to see it go every 3 months, though. I think that will make costs crazy. But yeah,
Brian.
Yeah. Dr. Kopp, what you're describing is something that stability testing will probe, and that's based on the storage conditions that the material is stored under over time.
Yeah. Thanks, Dave. Brian?
Yeah, not so much a legal point. Well, I guess it is a legal point, but I'd be curious to know how that will change, given the fact that I assume that some of this product is going to be transferred to a healing center, right? So then your storage regulations might change, or maybe you need to clarify like what happens to potency testing if healing centers are holding a lot of inventory for months on end. How do you account for that?
Then that seems to assume that cultivation will occur at a different place than a healing center does. But there is a possibility that cultivation could occur inside a healing center, in which case you could minimize transfer. But if we get back to that, the point is, I agree with what Davis was saying, is that we don't have the data yet. Stability testing will tell us that. I still think that we should be making a recommendation in the original rulemaking around how frequently retesting should occur, or maybe we don't need to make that recommendation. I know that didn't sound like a question, but it was. Duncan.
Can you make an initial recommendation that retesting must occur at a 3-month interval unless proven through stability studies that it can occur less often? I mean, can you make that caveat in your recommendation?
As far as I understand it, and Brian, I'm going to defer to you here if I get this wrong, is that the rulemaking is going to occur on a yearly basis, or recommendations around changing rules is going to occur on a yearly basis. Is that accurate?
You have to make recommendations. Yeah, annual recommendations. So.
Yeah, there will be annual recommendations from this board that could change the rules. So we could set a 3-month interval for the first year, and then realize that.
No. In your recommendation, could you just state, you need to do 3 months, unless you set up stability testing in your MIP or in your manufacturing facility or in your grow, and you prove to us with data that you actually only got to test every 6 months, because that's when you start to see the degradation that's appreciable, or in your stability studies you see it every 12 months, as Kim was saying. Can we make that as the recommendation for what would be the rule?
So Ken, I saw your hand go up first, but I'm going to go to Davis here, because I think that for the regulatory answer. Davis, what were your thoughts on what Duncan just said?
Yeah, I totally support that model, and we've adopted that same model in the cannabis space. We have a use by date that is 9 months, and that is unless you prove through stability studies that your product is shelf stable for a longer period of time.
Great. Dave, is it possible for you to share some of that language with Lori or Sam so we can review it at our November meeting?
Absolutely.
Great. Ken?
It's critical that we get actual data before we make a lot of rules. I don't want to require things every 3 months when we don't have data to show that we should. I think we should start out with maybe saying yearly, or if data show it should be, or to be determined as data show is necessary. I mean, I think starting with too many regulations is going to be prohibitive.
I hear your point loud and clear. I don't know that I would push out to a year. I think, if DOR settled on 9 months for THC, would there be, could we think about a 6 or 9 month window at least to start until we get some of that data? Like I said, Ken, we're really talking about the first year. So if people are providing us with that stability data and we see that a year is well enough, then we can make that recommendation after 1 year that we change the interval from 6 or 9 months to a year. So it's not going to be, I just want you to think this isn't going to be a rule that's set in stone that's going to live on forever. It's what's our starting point, and then what data do we want to see in order to go back and change it? And I'm not saying it has to be 3 months. I heard someone say 3 months an hour ago, and I just figured I'd start with that point. Duncan?
That's not to say that you can't run accelerated stability studies and get better data quicker than a year. So again, you can adapt your length of time based on what kind of stability study you set up. You can do the normal, just set it on the shelf and look. There's other ways to get stability studies that are accepted by the FDA and other groups that are accelerated. And so you are correct. I think that within the first year, I was just borrowing from the model that we follow, which is starts with 9 months. And then if you can prove you got 12 months or 18 months, then that's what you get to put on there once you have the data to prove it. And I'm not opposed to a 6 month or a 9 month. 3 months was just what was proposed in the document.
Yeah. Sue, I'm wondering if you had anything you wanted to add to this around stability. I know that you've done some work in this area.
Sorry there. I think you guys are right on track. It is something though, the FDA does require we provide stability testing in freezer, room temp, and refrigerator at 30, 60, 90 days. So it's quite expensive, but I'm sure the labs in Colorado can find ways to do it more cheaply. So, but I think you guys are on the right track.
And thanks, Sue. And again, the model here would be if we would suggest that every whatever the interval is, unless the cultivator can show. What's that?
1 year.
I know you're saying 1 year. I don't think 1 year is going to be our initial recommendation. I think that's too long. At least that's the way I feel right now. Unless you've got data that shows me that a year is sufficient and you can provide us that data. I'm a data guy. So if you've got data, I'm happy with it. Is there data out there that shows that 1 year is a good time frame? And what is that? What storage? Again, when I'm balancing this with this, are we going to make storage recommendations? Because I think if we're going to make storage recommendations, and I do think we should, then maybe the interval is longer. But if we're not going to make storage recommendations, then I think the interval has to be shorter unless the cultivator can show a longer stability through their own testing.
In the absence of any data, we're just picking a random number. I think we should pick a random number that is workable and easy for all parties. If potency is lost, a consumer will not get as much for their money, but they won't be harmed. There's not toxic substances.
Yeah, their wallet will be harmed, but not their brain or their psyche, right? I hear what you're saying, Sam.
So how do you feel about 9 months compared to a year? These are recommendations. Remember, we have to go through rulemaking where public comment is provided. That doesn't mean that we won't have data potentially in a month, 2 months, or 3 months, or when rulemaking, the rulemaking notice timeline that DOR has proposed is like last possible date that they will file for rulemaking regarding these specific rules, right, that pertain to DOR is March 1st, right? If we say 9 right now, that doesn't mean it's finalized even for the board. But if it is, we can also have that point where public comment can be like, all right, here's the research and literature that says a year and a half, actually, right? We just got, I think we don't have to set on a date or time, like just yet. I mean, we could say a year right now too. I just keep that into consideration that the recommendations aren't implemented rules.
Right. The reason I agree with you. I think that I'd like to make a suggestion because again, I like to hear what people have to say about the suggestion we're making, and maybe that informs better refine it. If we don't have any data at all, then we don't have any data at all. And the reason I push back against the year is if we're going to be, and maybe this is again, if I'm being stupid or wrong, tell me, but if we're in a window where we can change regulation once per year, then having a year means that window bumps up against it. But if we have a 9-month window, then we can inform it. And that's, again, as I say it, I realize it doesn't make a lot of sense. Strike everything I just said. Don't worry about it. Colton, what were you going to say?
I just wanted to input that I have done limited shelf life studies just based on room temperature, just out of UV light. So literally just in a styrofoam cooler. And there was pretty significant loss of potency at around 3 months. But again, that's why I was wondering if there was going to be storage recommendations, because clearly this was not the best way to do this. And that was kind of the point of my study in general, was I was just trying to see at the bare bones of how you may store these, what kind of numbers you might end up seeing.
Yeah, what I would love is because this is outside the bounds of my area of competence, and so I'm not going to pose this now, but before our November meeting, and usually we want this at least a week, if not 2 weeks before the November meeting, if anyone wants to provide draft recommendations for storage so that we can entertain those at the November meeting, that would make me incredibly happy. Not that the goal for this meeting is to make me incredibly happy, but it would help me out a lot if we could get some best practices for storage on the book, so we can start thinking about recommendations for storage. Sue?
Brian, I'm sorry, I might have missed this. So are any of the local labs there allowed to access the, fruiting bodies from the decrim market and just start some stability tests now so we can have data?
I'll defer to Sam or Brian on that.
I don't want to say anything or confirm anything or pitch any ideas at this meeting. Let me talk about it outside the meeting. Yeah, but I think
Duncan is smiling.
That might be an option, right? Like we just gotta see if it's not a problem on the legal side.
Yeah, so let's put a pin in that for now. I think what I'll come back with for the November meeting is probably a 9-month window as a starting point. Ken, sorry, it's not a year, but it's not 3 months. So we're going to go with 9 months and then I would especially for the non-voting participants who are cultivating, or anyone who wants to provide us with what they might consider good storage practices so we can start that conversation. I would really appreciate that. With that, I'm going to recommend that we take a break at this point and come back to pick up where we left off. So the board will take a short break. Please ensure all cameras are turned off and everyone's muted during the break. The meeting will resume at 3 p.m. Mountain Standard Time. So it'll take about a 10 to 11 minute break. Thanks, everybody. All right, as we're coming back, everyone just take a minute to reflect. I thought that was a really rich and fruitful discussion we just had. Really helpful for the board, I hope. And so thanks for that. Before we move to the draft recommendations for best cultivation practices, I just want to see if anyone had any other recommendations for testing or any other things they'd like to add. I don't want to spend too much more time on testing, but I just want to make sure we address if there's any other points that we should be considering. It's okay if nobody says anything here. Colton, I saw you unmute.
Yeah, I was just going to tell you that I unfortunately have to jump off here.
Oh, well, thanks. I appreciate the input you gave us earlier, and we'll see you next time, hopefully.
Yep. Thank you.
Thank you. Brian.
Yeah, so just a modification to number 3, the species testing. I would just add a sentence that says, if a licensee fails random species testing, the state licensing authority may order additional testing as necessary.
Yeah.
And that's not like ready for implementation in the rule. I think there's like some due process concerns and some other things that we need to work through. But I think as far as like a recommendation to the state licensing authority, at least flags it for future consideration.
That sounds good. Sam?
I was just going to ask if Brian could repeat that.
Yeah, so just a sentence added to number 2, excuse me.
Yeah, you can say you mean number 2. I think actually it's 2 or 3, right? 2 and 3. Because failed test, if it's either a failed species test or a failed test for other items, DOR needs to have a way to respond for a failed test.
Yeah, maybe it's just bullet point number 5, and it just says in general, if a licensee fails any testing, the state licensing authority may order additional testing as necessary.
Yeah, I think that's good. We'll just have it as an additional 5th point to refer to any of the testing, right?
Yeah.
Can you? I mean, I don't suppose you can fail potency testing, but I don't think we need to get that into the weeds on this either. So we'll just go with the 5th bullet point. Okay, so let's push on then to the next set of recommendations. Sam.
Oh, sorry, my hand is up. Let me just see where Billy is. He should be back.
Okay.
But Dr. Connor, I'll just throw it out there. You can fail potency testing in a, like, let's say an edible product or a manufactured product. If there were, psilocybin infused chocolate bars, a failed potency test would consist of more of a failed homogeneity or a failed target potency as determined by your SOP. So just a point to make about, manufactured products versus the actual just, fruiting bodies.
Thanks. And I do think we should clarify that the recommendations we've drafted so far, I think, are largely related to testing for cultivation prior to production of anything other than dried whole mushrooms, right? So we'll have to come back and make recommendations. That'll be our next step, is recommendations for testing after production or manufacture of a product at some point. But let's see if we can get through cultivation testing first, and then we'll come back to it. But a well-made point. Thanks, Davis. I appreciate it. Okay, so now we're going to refer to the draft recommendations for MGAP and cultivation for best practices. Basically, what we're saying is that MGAP should be used for best practice guides for small non-commercial growers, and we need to have more conversation and or determine whether we want to make a recommendation for actual size or leave in terms of what is a non-commercial small grower versus a commercial large grower. But we may leave that on hold for now until we get to the public comment or if DOR has any specific input on what a small non-commercial grower might be versus a large commercial grower. But that's just the language I settled on after the last meeting. But anyway, we would not require small growers to follow MGAP, but we'd say that would be for best practices. Large growers would be required to follow MGAP. And then licensed cultivators and producers who wish to produce forms of psilocybin other than whole dried mushrooms or powdered mushrooms, whether using some sort of extraction procedure to create chocolates, edibles, or other products, would have to follow food safety laws as stipulated by the state of Colorado. And then we would not allow for outdoor cultivation. And so these are the draft recommendations. And again, I'll open the floor for comment, questions, concerns about any of these recommendations. Everyone's so quiet. Ken.
Yeah, I have a concern that I haven't had a chance to enunciate yet. I think if we start making mushroom infused chocolates, that the feds are likely to shut us down. I think it's going to be a really big mistake to do that. And so I would just remove that from the language and just say edible, because here you're suggesting that this is something that we should encourage. And I think we need to be really shy on that, because I'm really worried about federal intervention once those kinds of products are available.
Can you speak about why you're scared that the feds would be mad if we made chocolates?
I think diversion. I think a psilocybin mushroom containing chocolate bar, one story of that getting into kids' hands or out of legal supervision is going to be a horrendous story that will cause harm to the entire industry.
And Ken, I will say that I think I've known since you first started attending our meetings that you have large concerns about diversion, and I have those same concerns. So I'm not opposed to striking chocolates out of the language. I just wanted to make sure that people understood that we were going to say that if you were going to produce some form of mushrooms that was food-like, even though I think mushrooms are food already, that you would have to follow food safety laws, because that reflected what we discussed at the meeting last week or last month.
Yes.
I agree with Ken. I would not put chocolates. I would maybe say extraction procedures such as edibles and or other infused products. It's a much more general language that doesn't invite, that heavy-handedness of like, oh, you're making candy, so therefore you're targeting kids, which is not the goal of any of this. But there will be opponents who will use that argument against you. We see it in cannabis all the time. And again, I do also argument the sentiments that if there is ever a diversion issue where somebody does get harmed who shouldn't have had it, it will be terrible for the entire industry. And so I'd rather not give us all a black eye before we even get up off the couch.
Yeah, I think that makes good sense. So we'll strike that language and leave it more general to say edibles and infused products. That'll be one update I make before our next meeting. Other thoughts or concerns about the size of grow, when you should be using MGAP, when you shouldn't be using MGAP, or any of those things?
We certainly would need to specify, because no one will want to call themselves a large commercial grower if you're subject to additional regulation.
Davis, you've been really helpful in this meeting so far, so I'm going to call on you again. Is there such a specification among cannabis cultivators?
I'm sorry, I got diverted real for choice of words, but yeah, I got my attention drawn elsewhere for a moment. Can you repeat the question?
Just wondering if there's anything in the med language around size of cultivators for cannabis growers.
No, not that I'm aware of.
So we might strike number 2.
We had a long debate about it, you and I, Ken, about whether we were going to require MGAP, and we said if it's a small grower, they shouldn't have to follow MGAP, and if it's a large grower, they should. So that's how it showed up. But if you want me to strike it now, I can entertain that idea.
Well, either that or we have to pick a size, and it's a random division. Do you want to say 20 employees? I don't know.
Right. The last time we kicked the number around, the number was 10 employees, and we didn't want to be too specific at that point. And I do think when we get to the final recommendation, we should be making a recommendation around what that number is. I don't know that we need to do that today. There'll be, Sam, remind me, there's a period of public comment before we go to final rulemaking or final recommendations, right?
There is a plan, to have like a listening session on topics like this. But DOR is doing listening sessions, so I encourage people to go there as well and provide these materials once you've made edits if you want them to include it.
Right. And so I think back again. Today we've drawn a lot of comparisons to cannabis, but we don't think about cannabis in the same way as we think about mushrooms in this instance, because you're not eating, no one's really eating raw cannabis, right? It's not something people would typically do, but people are going to eat raw mushrooms. We'll assume that to be true. And so I wonder though, Davis, in your experience, do you have ideas around large and small growers? Just curiosity as an employee of DOR.
Well, I mean, just in the MGAP guidelines, I mean, there are exemptions based on the scale of production and the vicinity of which you have your customer base. I don't know that that's directly applicable in psychedelic mushrooms, but perhaps we could draw from that as a model in some way.
Another thing to think about, rather than employee size, again, for the cultivators, is what about if we based size based on the amount of product you're inoculating per month or something along those lines, rather than the number of employees?
So some of the regulations are due to, are you harvesting every day?
Right.
And so that's, yeah, that makes sense.
So what if we based the regulations not on size, but on how frequently you're harvesting?
Some are based on physical size, like you need to have a bathrooms nearby, and you need to have somebody monitor. You need to have that bathroom cleaned every day.
Right.
Maybe you don't if it's just one person or two people. But anyway.
But you would assume some correlation between how frequently you're harvesting and how many employees you have, right? There's got to be some positive correlation there.
Yes.
Duncan, your thoughts?
I was going to say, yeah, we don't have the non-commercial and commercial grows, but we have tiered licenses on how many plants are flowering in the cannabis industry. And so we do some regulations based on size of flower, amount of flowering plants in a grow at any given point. And there is a tiered system at that in cannabis. So I don't, it's not directly correlative, but I do believe that you could do something based on either inoculations per month, if we're going to do a batch based off of that, or, amount harvested per month. And then that would also directly correlate. I mean, if you're harvesting every day, you're going to have more employees than if you're harvesting every other week, right? And so I think there will be some kind of correlation there. And then other non-commercial grows would be obviously religious exempt groups and things like that.
Yeah.
Would also probably, and we don't work in that area, but just as an aside to make sure that we're also thinking about that side of the coin, to make sure that that's included in the non-commercial grower idea. But that's my 2 cents. I think you do get correlation pretty good. I mean, as you harvest more plants or harvest more mushrooms, you're going to have more employees.
Yeah. So we could think about tying this to, the number of batches you're harvesting per month or something like that. Is there any data out there around? As I start to ask this question, I realize the answer is already no. So can you talk about these large agricultural grows like button mushrooms? Like how many mushrooms are they producing per month? Do you have any idea?
Oh, it's more like tons per week. I mean, it's really big numbers. It's hundreds of pounds per week easily for or even just for the specialty mushroom growers like oyster mushrooms and shiitake and stuff. For the button mushroom growers, it's tons per week. It's crazy amounts. Yeah, so I don't know that any psilocybin grower will ever get to the size that we're thinking about that would require all these requirements. I'm just not sure. I don't think the market will be that big in the state of Colorado that you will have that large of a psilocybin grower.
I tend to agree with you, especially from what we've seen from the individuals who presented us from the state of Oregon, where there's only a certain number of cultivators who exist now who've been licensed, who are operating in the regulated market, that we may not have, and they seem to have pretty small operations, right? They're 1 to 2 to 3 employees max, and that's including the people who are delivering or not delivering or whatever. So, so if we set a limit by employee, it still might be that because of that strong correlation, it might address it. I do think we should have an upper limit. And again, I can't anticipate what the future is going to look like, but we have some good estimations. I don't know. I'm still thinking about things. Other thoughts, comments from either board members or the non-voting participants or anyone else in the room?
I was thinking that perhaps fruiting space might be a good way to think about it. Square footage of fruiting space. The mushrooms need a very humid and environment to fruit in, and so generally a grower unless they're fruiting in mono tubs, will have a room or a tent set up to create that. And these large button mushroom operations, they have whole warehouses that are humidity and temperature controlled, where that they fruit in, in thousands of square feet. So somewhere in between might be a way of thinking about it, like how much space they could possibly be fruiting in. Because I think my concern about using the batch idea is, again, I mentioned liquid culture earlier, where you could inoculate, thousands of grain spawn off of one liquid culture. And if we're just considering that one inoculation as a batch, that could be a humongous batch.
Yeah,
that would take days to harvest.
Yeah, no, that makes sense. I do think that, how does the square footage argument strike people?
I think growers may never reach the size for the market.
But that's a good thing, right? But then that's a good thing, because then they wouldn't have to institute MDM. I think one of the things that I don't want to call anybody out, Oregon announced a different set of regulations. The one thing that was a little bit troubling to me in one of the presentations we heard was an individual who took things from their grow space into their house to wash them because they didn't have water access in their grow space. And I don't think we want to encourage that necessarily. At least I don't want, sitting here right now, unless someone wants to convince me otherwise. I think that you should have your own water supply. You shouldn't have to take your supplies back to your house to wash them. But maybe I'm wrong. Do you want to tell me I'm wrong?
If it's a single person grow, then why wouldn't that be allowed?
I saw Davis' hand go up. I'll think about your question, Ken, while I go back to Davis.
Yeah, so in kind of taking a look at the MGAP guidelines, there were also good handling practices. That is a sort of separate, more food safety oriented sort of standard. And perhaps I'm no expert on the good handling practices, but perhaps that's something that could be considered and or maybe already covered based on food safety laws in Colorado.
They're covered there, yeah. So Ken, to go back to your question, was the idea that the individual had created a separate space on their property where they were cultivating their mushrooms, and that was required by, I could be getting some of these details wrong, but I think they were required to separate the residents from their cultivation space, and all of that was occurring except for when they were going to wash things because they didn't have water in their cultivation space. They were taking the things to wash into their residence, and so it was this idea that they were commingling the spaces, and so that was somewhat problematic for me. If you're going to have a cultivation space, then even if it's on your property, which I have no problem with, and you're going to have a residence and the regulation separates them, says they should be separate, then I don't think you should be taking your tubs or your other supplies into your house to wash them. I think that's what my objection was.
Yeah, I understand that. So you could require plumbing and electricity, and certainly you need those things to be accessible. I just don't know how much regulation we're going to be doing around the physical grows. Do we need to be specifying everything? If this is, X square feet, you need this. Do we need to be doing that?
It's a good question, Ken. I don't have an answer for you yet. Duncan?
I guess coming from the cannabis world, everything's done within the regulated excise and licensing regulated facility. So there's no way we would ever be able to do something, even in our conference room, which is not licensed. So product never goes into our conference room, as an example, because it's not part of the licensed premises that's under video surveillance 24 hours a day. That's the other thing. So diversion things within cannabis, I can only imagine we should have another level because it's a psychedelic, right? Like protection. Cannabis is pretty, in my view, not as nearly as harmful or worrisome. But I would imagine that we have all of these safeguards and we are constantly going through excise and licensing and audits and all this stuff. I would imagine mushrooms would be on that magic mushrooms, psychedelic mushrooms would be another step up on protective controls.
I don't know, Duncan. I'm not gonna open the floor. I'm not gonna open the floor for that debate because I bet you'll get varying opinions about which one is more dangerous between cannabis and psilocybin, and I don't think that's what we're here to discuss. I do agree with the point that you're making, which is to say, if we have a cultivation space, then the products anything that's used for cultivation should stay within the cultivation space.
The licensed premises,
the
right?
Licensed premises.
Because I'm assuming excise and licensing is going to be licensing premises, and they're going to have their requirements for what your facility is supposed to look like on their own. I would imagine.
I think to that end, I don't think, Ken, to get back to your question, I don't think we need to specify all of those things because I think that's something that the regulatory agency can negotiate with each one of the growers or cultivators. Like, I don't think we need to specify square footage, things like that. But again, I'm just struck by the information we received in the last meeting where in Oregon, where they already have an established cultivation and they have video recording and all the security, and then, like I said, that they're carrying their tubs over to their house and washing them, and that just seems like a loss of chain of custody that I don't want to put in place.
True. Yep.
And so that's where I think the MGAP kits can be a little bit helpful because it can provide some guide for what people should be following if they're trying to set up a cultivation space.
When you go through MGAP, there are none of the regulations that you can make a strong case against. They're all a good public health. It's just cost. That's the only thing that if you require this, then you have to say somebody needs to be taking the temperature every day of this space and making. It's just lots of requirements. But
But we're in this space where we're saying we would only require that for people who wanted to establish whatever we determine a large grow is. And I think, Ken, you and I are probably in agreement that it's unlikely that that's going to exist unless we set that number at something ridiculously small. Like we're not going to say a large grow is if you have 2 employees, right? Or if you're harvesting more than, 2 days a month, right? But, if we're thinking about this idea of a large commercial cultivator, it's unlikely that that's going to occur in the first year, especially, right? No one's going to come here and set up a button mushroom farm to grow psilocybin mushrooms in the first year of implementation. And I don't know that it'll ever happen, but certainly not in the first 12 months, right?
Yep.
Yeah. And so that's what we suggested as best practices for the small grows, which we think will be what populates the state. And we require it for the large grows so that if there is a large grow, they would have to follow it precisely. So then we're back to the question at hand, which is what differentiates a small grow and a large grow. And I think that the fruiting space makes sense. Skylar, did you have a number that you put on that?
Yeah, I mean, the fruiting space could also be used vertically. So it might actually sound like a small square footage, but if you imagine using shelving as well, you could get a very large amount of fruiting space out of 500 square feet. Mm-hmm. I would say that would be a very significant fruiting space.
So anyone thoughts on imposing that as the requirement between a small and a large grow? Is there anything 500 square feet or smaller as a small grow and 501 square feet or larger as a large grow?
I would do it based on the volume of the amount of mushrooms that you're producing, like maybe 100 pounds a week or something like that. I mean, I think that's a better basis versus square footage, but I don't know.
Is that a good number, Ken?
I don't think anybody will get that high, but yeah.
No pun intended. Sam?
Yeah, it's like this square footage idea, I think, probably creates more challenges down the road if we're thinking of like licensed premises, if the healing center or is going to be cultivating or has a license associated with it and like the zoning of that. So like, I want to know, and Davis, I don't know if you can answer this. Maybe this is an answer for like Kyle and Dominique, or maybe even the folks in the cannabis space here. Or when starting, is there a business plan that's submitted on what operationally they're aiming for? I know like it's counted by like grow, but I feel like defining it is going to be different for mushrooms. so creating a restriction based off of size or size, or I guess what we would say is like batches or inoculants. I don't know. I think it creates some like unnecessary restrictions.
Sam, you're asking Davis a question, but I'm not sure I know what the question is, Sam.
I want to know if like when they're assessing like a large scale cannabis operation compared to like a smaller one, is it based off of just plants? Is like square footage taken into consideration? Is there a way we could do this in the first year where it's based off of, I guess, just like employees? If it's just one employee cultivating compared to two? I don't know, but I don't know.
Davis, are you still with us?
Yeah, I'm here, and I can't speak specifically to that in terms of at the start of a new business, whether their scale is predetermined or subject to different regulations based on what they expect. I will say that I think it's very common for cannabis cultivations to start smaller and work their way up in scale. But yeah, that's a good question, and I don't have the answer to it right now.
Thanks, Davis. Nathan?
I actually think it's going to be easier to do it based on square footage than volume, simply because, and cheaper too, because you can define the square footage of your grow space and where you're developing your spawn and all that, and that's going to be a fixed amount. If you're waiting to see how much product you produce, you could have, stumbled upon a great substrate mix and get more output, and then so that's a lot more variable. So you're going to have to be reactive to how much comes out, and then if you produce too much, then you have to, the regulatory agency is going to have to flag that and then go back and apply MGAP or whatever, or you're going to get growers that are like, oh no, I've made too much, and then destroy good product to stay under the limit. But if you set it based on the grow space, that's a fixed contained amount, and you have a good idea of how much you could potentially produce. And I think it tracks more with some of the regulations around bathrooms and things like that. So,
thanks, Nathan. Ken, I saw you on mute. Were you gonna, and then you muted again on me?
No, I'll pass. Sorry.
Okay. Again, I think it's good for us to put something in place so that we can look at it, we can discuss it, we can even get to the point where we recommend it gets implemented and then revisit it and see if it needs to change. The square footage model makes sense to me for the reasons that Nathan just put out, that you're less reactive and you're more in a stable environment when you're talking about the square footage space. And again, it allows people to make predictions about what they need to do and what the future holds. I don't know if 500 square feet is the right number, but that's the number that was given to us. So if we go in the direction of square footage space for a brief moment for conversation, what is that square footage? Is it 500? Is it more than that? Is it less than that? Go ahead, Ken.
Honestly, I would go with a really large number to start with. If you're going to do it square footage, I don't know what that number would be. But in terms of mushroom production, I would say something like 100 pounds a week. I don't think anybody will meet that. But if you were doing that, then I would say then you need all these things in place. I worry we're going to over-regulate at the beginning, and we can add regulations as they're needed.
Thank you. Appreciate it. Duncan.
I guess I could also propose something similar to the plant count model that we have in cannabis, which would be bins or bags per month, which is also a number you would propose to be a commercial level or not, which obviously how many pounds you're going to harvest, who knows, but you have a set number of how many inoculants you're going to make, how many bins you're going to do. That's very similar to how many plants we tag a month, right? So we have a certain amount of flowering plants. Once we hit 500 in flower, that's it. That's like our cap, right? Or 1,000, that's our cap. And then we move into the next tier, which requires another level of recommendation, another level of oversight. So you could do it if square footage is something that people don't like, or we don't really have an idea, then you can start thinking about how many bags somebody might do, or how many bins somebody might do per month, and get you to a commercial size.
I appreciate that point of view, Duncan, but we don't regulate the size of the bag or the size of the bin. And so, if we were going to regulate the size of the bag or the size of the bin, then that model makes sense, right? You know what a plant is, right? We have a good definition of what a plant is, but I don't know that we have a good definition. I know that bags come in different sizes. I know that bins can come in different sizes. And so unless we're going to regulate bag and bin size, that suggestion might not work, but I do appreciate it. Nathan?
I was going to echo that same comment, but also if we're doing it based on square footage, it's important that you limit what you're measuring, considering that a healing center could also have a grow facility on site. It should be limited just to the grow area, or maybe grow plus lab potentially, and not the conference rooms, not that.
Yep, yeah, similar to the way that the THC grows, where they have their cultivation space, right? That's Skylar?
Yeah, I think it's important to define that it's fruiting space, right? So it's like a humidity controlled environment specifically set up for fruiting, and that would be the space that I'd say 500 to 1,000 square feet. I'd be willing to double it and to say yes, that would be a really large grow operation, but defining it as fruiting capacity. The rest of the lab, they could be doing all kinds of stuff, and that has very little to do with actually producing mushrooms, and so just limiting it to that fruiting space.
Yeah. So having lived in apartments that were between the size of 500 square feet and 1,000 square feet, Ken, rather than 100 pounds, do you think if you set the limit to 1,000 square feet, can you imagine a humidity controlled fruiting space that's bigger than 1,000 square feet? For psilocybin mushrooms?
You don't. Yeah, I worry about this, honestly.
I can tell.
Yeah, I would go with 100 pounds a week, and then no one will get there, and you won't need to worry about it.
So the only objection I have, Ken, to your standard is that you're proposing a standard that no one's going to obtain, and I don't know that that's the way we want to approach this. Because if that's the way we want to approach it, then we would just strike the large grow language altogether, which is, I know, something else that you suggested. But I want to be anticipating if someone got to that size, right? And so I think I just want to have an.
I don't think the medicinal market can support it in a state.
I agree with you 100%. And yet I want to be prepared for it just in case. My motto in life is always be prepared for the worst case scenario. And so, if we're going to make the recommendation, again, we could entertain just striking the second part and just say mushroom good agricultural practices should be used as best practice, period, and just end right there.
That would be my recommendation.
I'll think about that, Duncan.
So when you talk space, right, you're talking a 500 square foot, but if I put 3 tiers in, I just tripled my space.
Yeah, that was.
Now are we going to measure the tiers and say, well, no, that's not 500, that's 1,500?
I think Skylar made that point when he originally suggested the idea of square footage is that you might think about the idea that you're going to be growing vertically as well as horizontally, and then you're measuring something different, square volume.
You're talking cubic feet, right?
Yeah,
at that
cubic
point.
feet. Yeah, exactly. But I don't. Again, so. Let's put a pin in all that stuff for a second and say, do we want to entertain Ken's suggestion that we just stop with MGAP should be used as a best practice guide, period? Do we need any other cultivation recommendations beyond that? End of my sentence. I'll open the floor there.
Well, to be fair, I'm with Ken on a lot of these things, less is more. And absolutely, if problems arise in the future, we have the option to put things in place. I think last month a staffer kind of said that too. It's well, if something comes up, we can definitely go back and try to regulate post the event. But
sure. Thanks, Skylar. Duncan.
Yeah, I go back and forth on it. I don't think within the first year or two anybody's going to hit that second point. But I would rather be proactive than reactive. if something does occur, I'd rather not have egg on my face. So I go back and forth, and I think we can always add later. So I wouldn't be opposed to removing it, but then I worry about the reactive nature of our world, and if we don't put something into place right now and we don't react properly, does it become even more regulated because now you have the outside pressures getting into the rulemaking time period. So I go back and forth. I wouldn't be against it. I don't think anybody's going to reach that commercial grow in the first year or two. So you'll have at least 2 rulemaking sessions before we might even have to address this, right?
Yeah. So Duncan, you just conceptualize why I lose sleep when I think about my work on this committee. Is I want to make sure we're doing the best we can without making some sort of big mistake. So I appreciate your comments there, Nathan.
I'm kind of fine with making it best practice, because if you're not following them on your own and you have bad practices, it's going to be self-limiting anyway.
Yeah.
So if you're trying to produce, you want to be as clean as possible.
Thanks, Nathan. Sue?
Yeah, I echo what everyone has said. Let's hold on. I think this was a really valuable discussion, though, and it shows us what we're. But yeah, they're not. That your fruiting bodies aren't going to thrive if you're not adhering to those. So I think we're in good shape for the first year or two, and we just keep it in the back burner if we need to implement it later.
All right. So if we go back to the one point that I was concerned about, maybe the idea that MGAP is best practice guide, and then we'll make a recommendation around how cultivation happens in the cultivation space, and that includes all steps of cultivation, including cleaning of the cultivations. I want to make sure that we're not allowing people to leave the cultivation space with product that may then. So Ken, this is the diversion argument, right? That I'm worried about things that might get diverted, but maybe I don't need to think too much about that right now. Duncan, I saw your hand go up.
I'm just thinking about the other committees. So you're going to have your safety committees that are going to make recommendations about diversion, right? And so other committees are going to address some of our worries. On the other side of things, some of those MGAP things are going to be addressed by building code, right? Like you're going to have bathrooms. So like that's one of the points in MGAP is bathrooms for people, but that also falls under other regulatory groups that are already going to take it. So using what we need to use from it, a good portion, there's a whole section about outdoor grow, which will be completely struck from the MGAPs anyways, because we're not outdoor growing as an example. So I think there's a lot less in MGAP that we should be worrying about, mainly because it's going to be taken over by other oversight groups or not even applicable like outdoor grows.
So Duncan, I'm on the safety subcommittee, so I have to think about it one way or another.
Oh, yeah.
So it's a valid point that there are other subcommittees doing work in these areas, and I'm on some of those subcommittees. I think, without having this conversation go on too much longer, I do have a hard stop in 22 minutes, and I'd like to look at the last 2 recommendations if we can. But I'll come back we'll come back in November with the MGAP as best practices and striking the rest of the language. I'll take the chocolates and other edible products out and just say edibles or infused products. And I didn't hear anyone objecting to the idea that we're not going to permit outdoor cultivation. And so I'm going to put a pin in that. The draft recommendations for labeling, I think at this point, what we have is all psilocybin products should be labeled with both psilocybin and psilocin content. And I'm going to ponder the recommendations that Davis made at the beginning of the meeting about how we might calculate that. I do think it's important to have both of those on the label and to have labels and provide data in such a way that the consumer can understand the potency of the product. And I'll add a recommendation around data potency testing so that we have this is the maximum potency and this is when that maximum potency existed, unless I hear objections to that. So your hand is still up, but I think that's a remnant hand, unless you were going to make a comment here. And then I do think with the best by dates, again, we can establish those. Right now, I think we're going to probably use the 9-month label as well, right? So if we're going to have a 9-month suggestion for potency retesting, then a best by date should probably be commensurate with our retesting for potency. Does that make sense to everybody? I know Ken's mad at me, but I'm not asking that question. I'm just asking if it makes sense. Right. All right, good. Go ahead. All right. So any other thoughts about what should be on the label? Total psilocybin, psilocin content, and like I said, I'll make a draft recommendation on one way or the other in the equation, but so that we have total potency as part of that, and then the date that potency was tested, and then a best by date. Any other things that we think should be on a label? And again, here we're still within this cultivation and sort of dried mushroom area. We're not talking about products that are manufactured yet.
So, in regards to marijuana, again, we have several labeling requirements that include perhaps not approved by the FDA. I can't even name them all. Keep out of the reach of children, things like that, to that nature. And so those are potentially things that we may want to consider requiring on our labels. Also, within the authority of rulemaking, we can work through that at DOR. So giving us the latitude to do so, I think, could also be a viable pathway.
David, I'd ask the same. Can you send that documentation to Sam or to Lori so we can review it in advance of the November meeting and see what things we might include on there? I think keep out of reach of children is a good one. I think other things for safety reasons are good. And so if you can send us those, we can review and see which ones we want to move over. I believe we'll probably also give DOR authority to change labels as well, but I think it'd be good for us to be comprehensive. Duncan?
So I actually have them right here. So there's this product was produced without regulatory oversight, health, safety, or efficacy. There may be long-term physical or mental health risks from using of marijuana, including additional risks for women who are or may become pregnant or breastfeeding. Use of marijuana may impair your ability to drive or operate machinery. Intoxicating effects of this product may be delayed by up to 4 hours. That's on edibles. And so not approved by the FDA. There's several terms, as well as the giant cannabis symbol that has to be a certain size on every product. And even if it's an edible, it has to be printed on the actual edible. Each dose must be labeled. And so just things to think about. I just happened to have it up, actually.
I appreciate that. I do think I want to remind us that we're not talking about a retail market, right? So we're not talking about someone walking into a store, buying a package of mushrooms and then taking them home. Yeah, that's not what this committee's entertaining. And that may tell us what we need to label and what we don't need to label, right? Because, again, my anticipation is that the rules, once established, will establish this chain of custody that facilitators maintain control of the product. It doesn't mean we shouldn't be entertaining some of these labels, but we may not need to be as comprehensive with knowing that people aren't going to be taking these packaged products home and then putting them on their kitchen counter or whatever. But I think it's important. Thanks for sharing those. Nathan?
Is there something that tracks with the training or what's required for facilitators? Things that if you look at what we say they should know or what they should be versed in, that's going to help inform what goes on the labeling.
That subcommittee is still developing regulation rules over that would be handled by the Training Qualifications Committee. And I'm on that subcommittee as well and can make sure that we're talking about that there. But I think there'll be some crossover where we have to think about it. The other thing to think about is what does the packaging need to look like? I know that for cannabis packaging, it has to be child-proof and it has to be really difficult to open. But again, if we're maintaining chain of custody, do we need that same sort of restrictions on mushrooms if the facilitator is going to be the one who's handling the product or keeping control of the product? So I think those are all things for us to think about, and we can come back to. But it sounds like at the very least we all agree that psilocybin and psilocin should be on the label. The testing date for when that potency testing was done should be on the label, and a best by date should be on the label. And the rest we can talk about at the November meeting if that's okay with everybody. And then very simply, draft recommendation for disposal of substrate, mycelium and mushrooms. All materials considered waste should be autoclaved and then composted. I know we'd had a lot of discussion around whether they need to be boiled, whether they could just be composted. And so I wrote this to be, I think, the most conservative of the way that we talked about it. I'm going to have us have a quick discussion about this before we wrap up for the day. So thoughts about the rule for disposal of all the products? Ken?
I would go with something slightly different. I think it's potentially expensive to autoclave everything. But you could have language that says waste should be rendered non-consumable and non-productive for possible further psilocybin production before discarding.
Ken, that was beautiful language that I didn't get. Can you repeat it or can you write it down and send it to Sam?
I can do both of those. I will repeat it right now and then I will send it. Waste should be rendered non-consumable and non-productive for possible further psilocybin production before discarding.
So we could give examples, so we could say this could be done via autoclave or by boiling or some other processes that...
Right, or compost. I think you should be able to take moldy mushrooms and throw them down the dish disposal, and grind them up. I think that would be fine too,
And I appreciate that, but again, I don't want them I don't want people taking them out of the cultivation space into their kitchen and putting them into their garbage disposal.
No, but if you have a kitchen that has a disposal,
agree 100%. That language sounded really good to me. Like I said, if you can make sure that the staff has it, I'll update the recommendations with that language, and we'll have a further discussion about that at our November meeting. So those of you that heard the language Ken just put forward, any concerns, questions, comments about what Ken just said around disposal? He said it much better than I did, and I like it. So, Brian,
Ken, one more time. Oh, yeah.
Oh, sorry, Sam. Brian had his hand up.
Just quickly, I would recommend, if you're going to use the word waste, I think defining what waste is probably a value add.
Yeah. And I can do that once I get that language. Once Ken sends to Sam and or Lori, I can add some definitions and some EGs, and we can discuss those in the November meeting. But yeah, I thought of that too, Brian. Thanks. Sam, what were you going to say?
I was just going to ask, before we hop off, Ken, one more time for your email. I just want to make sure we have it.
You bet. I will email it to you, but I can say it again if you would like. Waste should be rendered non-consumable and non-productive for possible further psilocybin production before discarding.
And it's a great starting point, and I'll probably build it up a little bit, Ken, and you can tell me how I got it wrong or messed it up at the November meeting. I'll try hard not to mess it up because I like the way it sounds. All right, I'm wiped out. So, Brian, do we need to go to executive?
No.
Perfect. So I'll start to close. The last bullet point is the next meeting time. Right now, tentatively scheduled for November 15th, 2023 at 1 p.m. Does that work for everybody? You just put that on your calendars as our next meeting. There's a note in the to remind everyone the next full board meeting is of the Natural Medicine Advisory Board will be held on October 18th, 2023 via webinar at 1 p.m. As I've already indicated to other individuals, I will be on a plane at that time, so I'll not be able to attend the meeting. I will send Lori an update of what this subcommittee has been working on so that can be presented at the full board meeting so that people understand the work we're doing. Sam?
Sorry, the error is October 20th, this Friday.
Thanks. Yeah, because that's 18th is today, so it's not today. Yeah. October 20th. Because that freaked me out for a second too, because I'm not supposed to be on a plane on the 18th. I'm supposed to be a plane on the 20th. All right, so the next full board meeting is October 20th, 2023 at 1 p.m. via webinar. And I'll provide an update of the work that this subcommittee has done to the program director so that we will be represented in that meeting. Before we get to the next piece about attestation and closing of the meeting, I just want to thank everyone for the work that we did today. I thought it was a really productive meeting. It was really helpful for me, and I really appreciate all the contributions from both the subcommittee members and the non-voting participants. And so I just wanted to give you my gratitude, especially also staff. I don't want to leave staff out, and Dora and DOR and everybody involved. Just felt like a really productive meeting, so I really appreciate it. With that, I would like to thank everyone for your attendance and participation during today's meeting. Before we adjourn, there are a few items to be addressed. As we noted, the next meeting, November 15th, 2023 at 1 p.m. Please remember your attendance is necessary to ensure a quorum for the meeting. I'll do attestations for the board members now. Do you attest and affirm that all conflicts of interest have been disclosed and the confidentiality of all board members has been maintained since the previous board meeting? I do so attest. Sue Sisley?
I attest.
Billy Wynn?
I attest.
Then I'll call for a motion to adjourn the meeting.
So moved.
And a second.
Second.
Thanks, everybody. See you next month.
Gotcha. Thank you.
All right.